Good morning, madam. Himabindu garu.
Hello.
Good morning.
Good morning, sir.
I think it's now clear. Voice is
Yeah.
clear.
Yeah. Are you able to hear me?
Hello?
Hello. Are you able to hear me, sir?
Yeah, we can able to hear you, madam.
Good.
And once your introduction is over from our side, your profile will be read. And next you can start your presentation.
Sure, sir. Perfect.
Just 5 minutes, madam.
Sure.
Thank you, ma'am.
Thank you.
Good morning, madam.
Good morning, sir.
Good morning, madam. మేడం మనం లైవ్లో షేర్ చేసి మాట్లాడాలి.
Video.
Good morning, madam. I'm the principal of the college, Srinivas Babu.
Namaste, sir. Namaste, sir.
Thanks for your acceptance and sparing your valuable time with us.
No, it's a pleasure as well. Thank you.
So you have to visit our campus in future, madam.
Definitely, sir. Definitely, absolutely.
And sorry for our reschedule time.
Not at all a problem. I get this.
We'll start it. Yeah? Thank you. Thank you, madam.
Sir. Thank you so much, sir.
Good morning, one and all. I welcome you all the participants to this faculty development program organized by Vigyan Pharmacy College in association with National Research and Development Corporation, NRDC. Today is the third day of the faculty development program, and today we have Ms. Himabindu, Managing Director, Novel Patent Services, Visakhapatnam, to speak on the patentable and non-patentable inventions and focusing mainly on the how to convert the thesis works into patents and the inventions for a COVID scenario, in the COVID scenario. I welcome on behalf of the management, principal, staff of Vigyan Pharmacy College, Himabindu Madam Garu, to have his madam's lecture today. Now I welcome Geetika, Assistant Professor of Vigyan Pharmacy College, to read out the profile of Himabindu Madam. Please, Geetika Madam, please.
Thank you, sir. Good morning, everyone. It is my pleasure to read out the profile of Mrs. A. Himabindu, Managing Director, Novel Patent Services, Visakhapatnam. Mrs. Hima obtained her master's in biomedical engineering. She was the university topper and is also the co-founder and managing director of Novel Patent Services, which is the only established company in the entire state offering unique business solutions in the domain of IPR with over 14 years of experience. Mrs. Hima is an alumni of Geetham University, Nalsar University, Indian School of Business, and Goldman Sachs 10,000 Women and holds certifications in IPR from Wipro Geneva. She is the only woman in business serving the IPR domain operating in the entire state. She is also the co-founder and managing director of Novel IPR Academy, the only IPR academy in the southern India. She was recently awarded with Exceptional Women of Excellence by Women Economic Forum at Hague, Netherlands on the eve of International Women's Day on 8th March 2018. She is a registered Indian patent agent. Ma'am, I welcome you to this EFTP program.
Thank you so much, Geetika. So, shall I take over?
You can take over, madam. You can share your screen.
Sure. Are you all able to see my screen?
Yes, madam.
Thank you so much.
You can keep in slide mode. Yes, madam.
Yeah. So a very good morning to one and all. So as the introductions are done, I'll just take over you to what actually is IPR, because patent forms a part of IPR. So I would just discuss what are the basic parts of IPR, and then jump into what is patentable and what is not patentable inventions. And hope you had a introduction of IPR, sir, or are the participants aware of IPRs? Thank you. Good. So just to give you an overview of what we do, because IPRs nowadays is perceived as the rights that we obtain, but there is a future career aspect as well which is associated with this. To be briefly precise with what we do and all, our company is a basic IPR boutique. We are associated with empanel analysis agents as well. Apart from this, we work with corporates, universities, research units, legal firms, independent inventors, startups, and people like this, where inventions come up, where inventions spring forth. So what we do, we have a 60 people, 65 employees who actually spend their time, their regular job is to work on patent analytics. So this is not new to India right now, because we are there over a decade. So we have engineers with us, we have pharmacists with us, we have engineers in different domains as well who can work with this. So it's also looked as one of the career opportunity, because IP with professional skills is a rare combination to be found with. So coming to what we actually do in patent analytics and all, I'll take you through the process. But before that, I'll touch basics of IPRs. uh, when you have IPR, it's a broad area. We have different sets, like we have patents, we have trademarks, we have copyrights, we have GIs, we have industrial design, we have trade secrets. These are the main 6 branches that we can talk. There are other rights called as sui generis, like plant breeders right and things like that. But majorly we'll discuss about these 6 rights. So when we talk about patents, patent is a right. All these IPRs are the rights conferred to the people who create them or who own them. As it's similar to a real property, you own a house, you invest in a house, you construct that, and you own subsequently. Likewise, patents are given as it's a right which protects your idea. It's not a mere abstract idea. We would discuss that when it comes to non-patentable matters. Likewise, if you have a name, some name that you register, some name that you want your customers to connect with you, some name you want to be, that you want it to be your recognition. So that can be registered as a trademark. And when it comes to copyrights, everything that we create, it can be a simple photography, or it can be a simple photograph, or it can be a manual that describes, or it can be a procedure that we write down in our labs, like for creating some, or for the process of making a drug, we just write down a simpler process, right? So whatever we write down in labs, when it is a procedure that we express in our terms, that can be copyrighted. So what not everything is a copyrightable matter. Geographical indication, now these are the rights given to a society for a particular thing which is popular in that area. And the special examples of this, as you all know, are the Darjeeling tea, the Assam tea, or the champagne. And these have the qualities associated with the products because they are coming from that part of the world. You have industrial designs as well, and this industrial design is given to the appearance. The Coke bottle, it specifically appears, though it is far from that we can identify this is a Coke bottle. So it's associated with the shape and aesthetics of the device. The other thing is called as trade secret. Trade secrets are the secrets, or we call in pharma, it's mostly known as the know-how process, right? You prepare a drug, and you add a catalyst, you boil it to you bring the solution to boiling at a certain temperature. You do not disclose the temperature, you only give them the ranges of the temperature. So these are called trade secrets, which are know-how processes for somebody, and trade secrets in another industry. Say, for example, the sales industry, the discounting procedure that we offer to the customers and all. So these are majorly called as trade secrets, which aid in business, but yet are to be kept confidential. Fine. So till here we are clear. And one more point, like all these 5 rights except GI, the patent, trademark, copyright, the trade secret, or the industrial design, these can be owned by either a person or a firm or a company as such. But this geographical indication is a right which cannot be owned by a single person. It should only be a society. Say, for example, you have Kanjivaram silks, it is owned by the Weavers Association there. Likewise, you have Tirupati Laddu. It is not a single person's asset. It is owned by a group of people, a society owns it. Likewise, another difference within these 6 rights is you have copyright, trademark, patent, GI, and industrial design. All these 5 should be written, whenever you want the right on this, you have to claim the right by disclosing whatever you invented or whatever you believe that to be your asset. But for trade secret, we do not disclose it to the public. It is kept confidential with us, yet it has a right called as trade secret. And if there are any questions, I think we will give you some time after that. Probably you can start pinging the questions there. Now coming to a few important aspects, though it is patentable matter, one of the hazards of trademarks is what I'm going to discuss. Trademark, it's a name given to, it's a the right given to a word. It can be phrase, symbol, design, or combinations, or it can be smells, of course. It can be music compositions as well. That is given a trademark. And the basics are these 3. It is called as TM. When it is unregistered, you have a name, and on top of that you'll have a TM to specify that this mark has gone and is an unregistered trademark which has been filed with the trademark office, but yet is waiting for its confirmation, like grant. Now SM is the mark registered for, it's an unregistered, it's called as service mark. And when these rights are registered, like granted, then we use R, which specifies that it is a registered mark. So like I told you, letters, numerals, colors. So there is a famous, quote by a famous author called as Shakespeare. So he tells what's in a name, but his own website now costs more than 1 million USD. And whenever companies are valuated, mind me, whenever companies are valuated, valuation is the thing that we do when we sell the company, when we sell a product, right? So when brand is created, the trademark also contributes to the valuation. It's not merely a product that you sell, it's not merely a company that you sell. There are trademarks that also will be valuated. For example, good night, right? Good night product is there, the solution, or there is a secret formula where the mosquito repellents are being constituted. When this is being sold, this good night actually is not owned by Godrej initially. Godrej bought good night, and when it bought good night, it only had bought the trademark of good night. It did not buy the entire building, nor it didn't touch the raw materials or anything. It was only the trademark that Godrej bought. So this is the value of trademark. Another thing, the franchises that we give. Whenever somebody manufactures or somebody sells, or the schools, for example, the Kidsy, or the franchise like food outlets like the KFC or McD. So all these are franchises which are liaising based on the trademarks. So this ISO, there is a certain difference when it comes to ISO, FSSAI. Right now, these are being the famous trademarks. So these can be owned. These are called as certification marks, because these certify the quality of other goods. Collective marks, like you form associations, you can give a collective mark. Why should we learn trademark? It is a patentable, non-patentable matter. How does it relate to you? These are called counterfeits. N number of varieties that come in India whenever you search for products, right? So these are the different ways where you can Have you have these kind of stores at your places? The original is this, but we have many coming up like this, right? Even with Nutri. So what happens to medicine or pharma industry? This is the industry which is most prone to infringements. There had been many, when customs do the duty, because we are part and parcel, like we also work with the customs department here. So what we do whenever the shipment comes, there are many tablets like this, which are not originals. These are fake. These have wrong individuals, wrong ingredients, but they are therapeutically active, but not as active as the original thing. There can be side effects as well. For example, you have them in medicines, you have them in what do you call, toothpaste, the baby foods and all. So to stop such kind of importing things to India, we should have an idea of the trademarks in order to stop the imports. So that's the basic crux, where medicine has a very beautiful link with the trademark. And this is an ongoing infringement, mind you. The clothing industry, the automobile industry, they only contribute to 20% or 28% of the overall infringements that happen from the customs. So now coming to our topic, patents. So what do one understand by patents? You can just unmute. Any participants, if you just wanted to talk, please unmute, or you can raise the hands. So at least an idea of what patent is, or what does it offer. Any idea on patents? Have you ever, any the 32 participants, did you ever file a patent, or did you ever apply for a patent? Did any one of you apply for a patent? Please raise hands. No? See, patent is the simplest thing. You might have worked in, yes. Very good. So, Mr. Ghosh, Deba Prasad Ghosh, very good morning. So, you have applied for one formulation patent, right? Can you please share what was your experience? How did the process go? I think you can unmute and you can just share your experience.
Hello.
Yes, sir. We are able to hear you.
There are certain this patent officer is there. He's also from a company.
Wonderful.
So he came and he just checked our patent application. This application first initial search. First they have done it is an initial search for whether our formulation is patentable or not.
Absolutely.
Then we have just prepared a one draft for our patent and submitted to him. It is under his review. Afterwards, he will report us back that when we can file this patent.
Wonderful. So, any other participants, sir? Like, what do you think is the importance of having a patent, sir?
So important is that our work will be safeguarded, because if we just publish before patenting, then if it is something useful, then anyone can copy our work. But after patenting, our work will be safeguarded, and if someone wishes to utilize our work, then he or that company has to take our permission or take our license from us to do this in industrial scale or do future work.
Absolutely. Absolutely. Thank you so much for your input, sir. So, why are patents, or why do people invest their time in creating something or developing something? What's the main basis that they do this? Why should they ever invent or work on some subject? The main thing the first thing that comes into picture is the first and foremost thing. 99.9% of the inventors, actually, when the research is done to understand the scenario, especially in Western countries like US, 99.1% of the inventors said because of monetary benefits. Absolutely. Because your inventions reap the mounts. That's so good. What is the second thing that people look for? To safeguard or to own the name or to have the credit of inventing something. So how is that taken care of? To have the credit of inventing something, there are Wright brothers who took the credit of inventing aeroplane, or there is Thomas Edison who took the credit of inventing a light bulb. But how many of the experiments which the other scientists actually have come up with, that has not been in light? The other thing, Singer sewing machine. How many of you think that it is a property of Singer? Do you know that it's not a property of Singer? It was some invention which was initially invented by some other scientist, some other person, and Singer copied this. It took for an infringement. Copying in IPR is called as infringement. And when it did copy the invention, it knowingly did this because it wanted to have a licensing agreements. Usually there are strategies where people ask for licenses, and this is one of the strategy that Singer took. So this is how actually people come into picture, or IPR issues come into picture. So the other thing is securing of rights. As Sir told, most of the academicians, most of the people in academia would like to contribute. It's not monetary benefits that most of them look. It is only to contribute to society. So here comes contributing to society, which also happens to be one of the motive behind your researches. The other motive, that most people have is staying ahead in the niche areas. Say, for example, there is now some calamity or something coming up in the society, which has to be addressed. For example, now it is COVID. So when COVID is addressed, most of the inventors come working on this COVID scenario. Likewise, you have many problems which are being addressed in one of the niche areas. So all these also is one of the passion. And the other thing, having fun. Most of the scientists have fun only till inventing, but not to the game. They can't go beyond the game of claiming the invention, or working the invention, working out, reaping the benefits, and utilizing the IP. This is something which the business people do, right? So having said that, patents basically protect whatever we do. They can be both product as well as process patents involved. And the term of patents is usually 20 years. And the other thing of the patent law is examination will be done only on request. Here, what we do is, when I take you through the patent filing procedure, I'll explain you all these three. But in general, the IPRs are only for a certain period of time. For example, patent is live only till 20 years. What happens after this 20 years is the public can use without seeking the permission of the inventor. The public is free to use. Anybody is free to use after 20 years after you get a grant of patent. Fine. The other thing, but before that, before going further into patenting system, I would also like to give you three important things which qualify an invention or which qualify an idea to earn a patent, right? So whenever invention, we call it an invention, means it means that either it is a product or process which involves an inventive step. So whenever we say that it is invention, it should be an inventive step which is associated with this. For example, now we invent something in India, but what if something is already there in China? And all these IPRs are geographical rights. Means if I want to exercise my right in India, I have to file my patent application in India. If I want to exercise my right in US, I have to file a patent application there in US. And likewise, in any country, if I do want my patent to be enforced, enforcing means I want to I want to stop the third party from using my invention. If at all they use, as sir said, they have to seek permission from the owner of the patent in order to use. If they use it without seeking permission, that is called as illegal use, and that is what is called as infringement of patent rights. Fine. So whenever infringements happen, this is how they happen. They use the invention without seeking the permission of the owner, and that is legally not advisable. They can, they can be sued, or they can be brought in front of the court. So this is how it happens. And when it comes to inventive step, and as I said, IPRs are geographical in nature. Point one, it should have an inventive step. Though I file it in India, I would claim that I am not going to sell my patent in countries like America, or countries like China, or countries like Japan. I am intending to sell my invention, what I call it as invention, only in India, and I file it in India. Then what will happen? Sometimes, in the first process called as patent search, if they find out that similar invention is there filed in China, and I argue that it is not in China that I have to, market my invention, it is only in India, and India doesn't have a filing, it is still invalid, because patents are granted only to invention. When something is filed in, the same thing is filed in some other place, it will be called as discovery. Discoveries are not at all given a grant on patents. However, you can file the patent, but the grant will not be obtained. This is the only difference when you file discoveries and you file inventions. And what is new? Invention must not be anywhere. The first thing, it should not be published anywhere in India or elsewhere. For example, the thesis work, whenever we want our thesis to be patented, the first and foremost criteria is we should not give a paper publication. Here also, most of the inventors, we work with many of the Indian domestic universities as well, and we have filed in AP itself, we have filed more than 300 patents pertaining to, many of the academicians or universities. Whenever we work with them, for most of the people who are not aware of IP, they would have said that they already published it somewhere. And when we just dig out what they published, usually, they restrict themselves in applying for a patent, believing that, this rule is violated. We have published, madam, so what do we do? Though you have published, there will always be a possibility where you don't publish the actual process of how it happens. You might disclose them only the advantages of that. The advantages of the invention only must have been in 85% of the cases. We found that it is only advantages that were published, and in other 5% of the papers, what we found it, segregate your the publications in 4 or 5 papers. You don't publish actual thesis into whole one paper. So this gives you a window where you can flexibly add on something, the 5th part that you segregate, that can be guarded as a patent as well. So that possibility also will be there when you find out or when you want to publish a thesis. And what is the actual what do you say, what is the favorable time to convert an idea into a patent? What would be the favorable period for anybody? The main thing is this cannot be decided. For example, it always depends on what's your use. How are you going to utilize this patent system? For example, people who have the idea of adding it to the resume, you can on day one, as soon as you have an idea, you can convert it as a patent. It does no harm to you. But if you are in business, or if you wanted to like if you wanted to tie up with somebody, or want to give it to somebody, or want to do a business on some patent, or want to commercialize, is what we call, okay, commercialize some patent application, then it is better to have time, because I already said that patent is valid only for 20 years of time. So you have to search for a suitable person or suitable investor who will buy, or suitable company who will adopt your technology. So if that groundwork is done, then it is a suitable time for you to go for filing. And this also has its own challenges, in the sense, though you want to pay, you want to patent it later on, you might lose your priority. Priority means reserving the invention on our name. Say, for example, inventor A is there, inventor B is there. Inventor A, both of them pursued an idea. They got an idea and they wanted to go for a patent. Now, inventor A thinks that it has to be fine tuned. Both of them got the idea, conceived the idea, say, for example, on Jan 1st, 2020. Now, this inventor A will think that I have to work it on further, I have to refine it, I have to define the procedure, I have to have a prototype, and in pharma, I have to elaborate the process, or I have to show how this is done. If it is a BBB kind of barrier, or BBB kind of, in a film, then I have to demonstrate the efficacy of this BBC, and I have to demonstrate that the drug can pass through this BBB, a carrier or something utilizing this. Then this A will sit on working further on that. But what the B does is, the B will immediately go and file a patent immediately, and then he will start working on the product refinement. So though A achieves the product refinement earlier than B, but he will not be awarded a patent, because, and he can't market the invention also, because he has got no patents. So in order to market, in order to give it to anybody, in order to import, in order to export, one should have the patent rights. So this is how the limitations are there. So as soon as you conceive the idea, we can reserve that. For the ones in business, we also have another option called as provisional patent, which we play with. So coming to the inventive steps and all, so it should have a technical advancement on the existing knowledge. So that is one more reason why we perform the patent search. We perform patent search to know whether this is the first of its kind. The second thing, what was the prior art? And in compared to the prior art, what's the advancement that people bring in? So this is the normal procedure, how the patent filing will be done. We, when it comes to, the people like us, like the consultants, or when you give it to an RDC, or when you give it to us, what we generally do is, we generally do a basic preliminary patent search. After search, we draft the patent. And when we draft the patent application, we prepare the draft, we draft it, and then we file it with the patent office. So from here, the original government procedure will start. We file a patent application. This patent application can be provisional or can be complete. Provisional is an application where we file the abstract of the actual idea. It is not the complete patent application that we file. It is only the abstract. Means it will give the inventor some time, point one. The other time the inventor gets it, he can refine his idea. He can make improvements on the idea, and he can come up with more better solutions. So it is the provisional that we file for giving them more time. And if he is ready with the complete invention as such, then we go for filing a complete specification. And if in case a provisional is filed, then within 12 months, the complete specification has to be filed. Else, your patent, which is filed as provisional to earn a priority, is void. It will not be considered as a patent. After 12 months, if a complete specification, if it is not filed, then please presume it that you did not at all file a patent. If you don't file a complete specification within 12 months. Then what will happen? Anybody can file. Once you file the provisional on Jan 1st, within 12 months, within December 31st, if you didn't file a complete specification, if there is another inventor who will file the same matter, say for example in June, he will get a patent if he files a specification. Fine. Then the other thing is publication. Once we file, then government will publish it in the government journal, right? And in this journal, it is called as publication of patent. And this application publication will happen after 18 months after we file the patent. You can also prepone it. In the sense, there is a fast publication option that is available in India, where if you file, if you pay a certain amount of fee to the government, they will publish your document within 1 month as well after we pay the fee. The other thing is request for examination, and once after this publication is done, you have to file a request for examination. Now, how is this important? In the sense, only when you file the request for examination, your application will be put for exam. It will be going through, the examiner will be going through the application, and after going through the application only, the grant phase will come, if at all it is a grantable matter, right? So if request for examination is not filed, then your application, though it is novel, though it is commendable invention, grant will not be obtained. So request for examination is a mandatory process in order to get a grant. So when is this filed? There are certain timelines. So it should be filed within 48 months after your filing date. And what happens if you don't file? The application is published, it is laid open for anybody to copy, and there will be no infringement if something is copied after publication and request for examination is not filed, because it will be abandoned, the application will be abandoned, and it will come into public domain. So anybody are free to use this. The other thing, once we file the request for examination, they will give the first examination report, and once the first examination report is given, the examiner gives the first examination report, and we usually respond to those objections, certain objections or clarifications that the examiner seeks. And once he is satisfied with all the objections, he will grant the patent. Fine. And after granting the patent, 20 years of time, we have to just go in paying the maintenance fee. If we leave the maintenance fee, then also the application comes into public domain, and anybody can use it here. And certain patents, especially in pharma, once there is a thing in pharma, many of you have heard about generics, right? So what are these generics? Any idea? Please raise your hand so that I can just see how many of you have the idea of what generics is. What are generics? We hear this a lot in pharma. How many of you have the idea of what generics are? Sorry?
It is generics are off-patented molecules. That means already innovator has done, and which is already patented and it's expired.
Absolutely true. But how can generics be when it is expired? I have a simple question here. When you are saying that it is not all can manufacture the things, it is only the patent owner has the right to dictate the market. Then once the patent has expired, then why only 1 or 2 companies come up with generics? Why not all?
They may have taken the permission from the innovator and modified a slightly formulation or the physical or chemical attributes of the main component, drug molecule. The original process is of the innovator. The original molecule is of innovator.
Absolutely.
But they may have changed the polymorphic form of drug molecule or any packaging technology or any drug substance components. Something they might have changed it.
True, sir. But my question here is, see, how long does the innovator have the hold on an invention? Only till this 20 months. Once the 20 months is done, the innovator or the patent holder will not have the hold on patent. Means there is no right. It is a void. Anybody can use. So innovator also will not be able, in the sense, you claim a property and somebody write that you own this property only for a limited period of time, only for 20 years of time. Now, after 20 years, in the 21st year, if I come and ask you to legally authorize me to stay in that property, you are not the owner, because your ownership is only for 20 years. So after the commencement of the ownership period, how can the innovator grant somebody to license or grant somebody to manufacture the generics? And absolutely true, because generics are non-patented products, right? Generics are not patented. They have changed the formulation. Why then, if at all they change the formulation, then again it is new. The formulation is not the same. Then why is patent not obtained? Point one. And how does the inventor claim or have any command over that invention or over that formulation? The other question is, why are generics not being manufactured by all the other companies? There are only certain companies that manufacture generics, not all, for the same composition. Why is that so? Can't they, replace chlorine by fluorine and launch their product? They can, actually. Generics are that, right? But why are they not modifying it?
Ma'am, they have to perform this bioequivalence study.
Absolutely, sir.
And thing is that they have to purchase the license from the main patented company that we want to produce it here. Like where I used to work, they used to produce some generic drugs for this GSK and other companies.
Mm-hmm.
So after this patent is over, then regulatory affairs department is there, which keeps an eye on when some patent will expire. And
Mm-hmm.
after that, they can start the production of that particular molecule after doing this bioequivalence study that their formulation is as effective as the previous one.
Absolutely. So that is how generics come. You have
And
said half of that is true, but who will, when the patent is expired, it comes to public domain. Anybody can practice. Say, for example, you have Flatron's TV, sir. Nowadays, Flatron is visible everywhere. But before 10 years or before 20 years, Flatron is a very rarely seen quality or of a TV. Because the patent rights have expired, everybody can use it right now. Likewise, when patents are expiring for a drug, why is only one or two companies showing the bioequivalence? Whereas there are many companies who can prove the bioequivalence and jump straight into manufacturing of the generics. For example, if you replace something by chlorine, and if one of the products launches it, the other company has the same possibility of replacing it by fluorine, yet showing the bioavailability and jumping into the market. But why is it not being done so?
other companies may be working because if you say 10 years is a long time, other companies may have invested in other drugs,
Mm-hmm.
which with which molecule might be far better than the this one.
Mm-hmm.
So 10 years is a long time, and 10 years it takes for new drug this development.
Hmm.
Another thing is that although patent is expiring, they can also extend the patent. The
Okay.
main company can extend the patent, and so again that second company, loan license company, has to obtain a license to manufacture that particular drug here.
Okay. So one of the possibilities, you say that patent extensions can be done. Second possibility is, might not all the other companies would have the interest of manufacturing this particular drug. The third thing, as it is a new drug, it will take minimum of 10 years to establish the bioavailability. Right, sir?
By
That is
modifying
that the 10 years new drug molecule may appear in the market.
Okay. Okay. So another.
slight modification of the original molecule,
Mm-hmm.
I think may not get the patent because the therapeutic efficacy of the original inventor molecule and this molecule will be same.
Absolutely. Okay.
And we can change the molecule. If we chemically modify the molecule, then it will be entirely new. We have to again do the study whether it will have any bad pharmacological adverse effect or not. But we can change the formulation only.
Okay. The other possibility of generics, sir? So these both of the statements, like whatever Deva Prasad Ghosh sir has given, and also from Vignan sir has given. So I'll clarify both of them. But before that, there is a slight modification that the drug approval process also comes into picture here. Whenever any industry has to flourish, except for engineering or a certain exceptions are there, but mostly when it comes to drugs, medicines and all, there are regulatory boards that come into picture. For example, IND, the investigational new drug, as sir said, it definitely would consume more time. 10 years is a very less period of time, sir, but mostly when it is a new drug that comes into picture, minimum of 13 to 14 years of the time is taken to establish the clinical trials, the bioavailability, bioefficacy, all those studies are being done to establish the safe, intake of that particular drug. And this is the minimum time that is consumed for the R&D, research and development, that goes in, right? And if patent term starts in the year one, the moment they consume or they conceive this idea of patenting this particular drug molecule, they file a patent application for the drug molecule. They keep it aside. Now they will work on the R&D, and after 14 years, finally when they complete their research and finally when the drug approval is given by the FDA, they launch that into the market. So what happens when they launch this into the market? What will happen is the effective period where they can sell the patented molecule will be roughly 6 to 7 years, because 13 to 14 years is consumed in their research phase, R&D phase. So the effective period will be only 6 to 7 years. Only in this 6 to 7 years, they will be able to market their drug. That's it. The other thing, there are various processes like NDA, new drug application, is when, we process it there. There is another drug application called as ANDA, abbreviated new drug application, right? For this, this also, preclinical and clinical data that establishes the data, safety and efficacy, these also have to be done. Apart from this, there is nothing in India. There are, as sir said, extension of patent term. This is not there in India, sir, anymore. We don't practice it. Whereas in countries, in few of the countries, still, if it is a IND application, if it is a new drug entity, NC, new chemical entity, for this there are certain terms where the extension of the drug will be given. Apart from this 20 years, 5 years exclusivity, or if it is an orphan drug, there is a certain time. If it is a pediatric use drug, there is a certain extension of time. But in India, we don't have that extensions anymore. Fine. So what will happen? The other thing that people have to know is clinical studies. There is, for example, Pfizer came up with torcetrapib, and they actually started manufacturing of that, and they applied it. It was for treating the cholesterol, hypercholesteremia patients. So what happened was they invented the drug. They had a patent for that. Patent grant also was done. It is a very good new entity. And parallelly, they have conducted the clinical trials. And after conducting the clinical trial, one was successful. They proceeded with clinical trial 2, phase 2, where more than, the total study has involved more than 7,500 patients were involved. And more of the manual powered, like manual, what do you say, help was also taken for this particular drug. And they prepared, because once the clinical trial 2 almost, they turned out to be a positive thing. So they planned a factory in Ireland. They have invested, say, for example, 1 billion investment was drawn to build a nice factory and all. But in New York Times in 2006, they recalled all that investment that they did keep for this particular drug, because the clinical phase 2, it was a failure. Though they had a patent, the regulatory affairs did not allow them to produce that into the market. So though they had a patent, it was a failure drug, so they have to take back. So in pharma, the costs in investments that goes in R&D is very huge, and there is a limited time frame that they can really market the drug, because it is only in the term of patent that they can actually market their drug, which is effectively only 6 to 7 years. That is the reason why the costs of the antibiotics or few of the medicines are very huge. So to beat out this, there are generics in India. Whenever we talk about generics, as you all said, generics are non-patentable matter. Why are they non-patentable? That we can discuss in this sections in the patent, in this presentation. Apart from that, generics definitely are not patentable, point one. And generics are the, like they modify the actual composition, they modify the actual chemical entity, and it is not the original entity, original drug molecule that is applied for a generic. And in generics also, it is only the bioavailability studies that has to be established, that's it. And if we establish that thing, then the rights will be offered by the regulatory boards. Here patent board will not come into picture, patent system will not come into picture, because once the patent is finished, the term is finished, it's finished, it's done, that's it. Generics, we don't award the patent for generics, point one. And when generics come into market also, the drug regulatory affairs, that boards will deal with all this. It is not the patenting thing that will come into picture. And why is that the generics are not patent? In the sense, in pursuit, like when Indian Patent Act of 1970 is taken, the section 3 describes some of the exclusions from what can't be, because what can be patentable, everything can be patentable, except this is what this section tells. So if we take, there are certain exceptions, like frivolous inventions, the inventions that lack utility, or where though the inventions are patented, it does not actually serve the purpose of benefiting the public. For getting a patent granted, novelty is the first thing that we discussed. The second thing is the obviousness. To get a grant on patent, the second thing is obviousness. It should be non-obvious, it should never be obvious. Where generics come into picture, where fluorine can be replaced by chlorine, there will be certain ifs and buts, but properties remain almost the same. Or obviousness, somebody invents, the famous example of Kapo noodles. The person who comes up first, Kapo noodles, with Kapo noodles where instant noodles, the water can be poured inside the packaged thing where the noodles cooks itself, and somebody else will come, you have given a patent for Kapo noodles, I'll prepare a glass of noodles, nobody has used a glass with noodles inside, so this is my invention. And he comes up and he asks. So obviousness, it is quite obvious, the one who builds up in cup can also obviously be using it for a cup as well. So obviousness is also one of the concept which rules out the novelty, or which rules out the grant for a patent. So the invention should be novel, it should be non-obvious. The third thing is, invention should be applicable, industrially applicable, it should be benefited to the mankind. So this is one more criteria that people see. And such things when seen, frivolous inventions, like it's also called as unnatural invention. Say for example, I give a drug which can cure cancer 100%. There is no such drug, it's a frivolous invention, and contrary to the well established natural laws. One of the things why cloning also can't be, it is contrary to the natural laws. The other thing that is accepted is commercial exploitation of primary use of invention, which is contrary to public order or morality. The gambling machine, it is contrary to the public morality. The government doesn't agree for this. So any patents where the inventor does something to the gambling machine and comes up with that, it is not patentable. The second thing, famous example is, I develop something which is very much useful for the thieves to theft some property. Those kind of things are also not patentable. The house breaking devices or something like that, they can't be patented. The other thing is where it causes prejudice to the human health or plant welfare. All such inventions are not patentable. For example, we have biologic warfare materials or biologic warfare devices. All these destruction weapons, though it is given to Navy, or there is certain atomics, these invention also accepted to be granted a patent. The Terminator gene technology, we don't have a patent awarded to this because as per our patent law, it is not patentable matter. The embryonic stem cell culture, all. And the other thing is mere discovery of a scientific principle. All the mathematics people come here. We just can't restate the Newton law, or when they say that E is equal to mc square, all the abstract ideas or the scientific principles, or the calculative methods, these can't be actually patented. The other thing, the mere discovery. So this is the important section which I was talking about. The mere discovery of a new form of a known substance, or which does not result in the enhancement of a known efficacy of that substance. It can be the salts, esters, ethers, polymorphs. It can also be metabolites, pure form, particle size, isomers, mixtures of isomers, complexes, combinations, so whatnot. So if we just change, the generics are these kind of things. They are not very new. It is a mere discovery of a new form, though it is true, it is a new formulation, but it is a known substance. Only few of that original molecule are being replaced, but it is a new form of a known substance. So all these salts, esters, that is the reason why they are not granted a patent. Howsoever, you have a, say for example, we filed a patent for Shankar Eye Foundation. Their R&D team once developed a coke crystals. Here coke crystals also will be there. Esters, ethers, all these pure form or particle size. Once we developed, crystalline forms of known substances are not at all patentable. There is a drug called forskolin, where this is put into the eye of the patients, where ophthalmological procedures are being done. So what happened was, this Shankar Eye Foundation people, they realized that it is quite irritating when it is put into the eye, the forskolin. So because it is a crystallizable form, and these it is some roughness is created when you just drop your this one eye. So what they developed is, they developed a crystallized, like forskolin crystals. Now these forskolin crystals, when put into the eye, so when these forskolin crystals are put into the eye, these are less irritating. These are not causing irritation, point one. And the efficacy of, or the solubility of forskolin within the eye has also increased. And when we have demonstrated that it is more than 85, when enhancement of efficacy is there, there was a patent which was produced similarly in electronic domain, where the cost of the product comes down by 1 rupee. No patent will be granted, though it is a known substance, though you are enhancing the quality, though you are enhancing though it is less, you are reducing the cost of the product, but it is only 1 rupee that you are decreasing. So that doesn't mean to be a larger difference. But for this forskolin, it is almost 85 to 90% efficacy of the substance. When it is in the crystallizable form, the absorption has increased to 80 to 95, 90%, 85 to 90%. So though the 3D says that we can't award a patent, but we had the honors of getting a patent granted because of this thing. It was a crystallizable form. This was an exception to the 3D. So not all co-crystals, or not all esters, or not all salts cannot be patented. There are few things that can be patented, and how the exceptional, the way you draw the exceptionality also differs. The way you draw out the non-obviousness also defines how you can get a grant on these patents. So this is one of the examples in contradiction to this 3D that we have. The other things are the generics. Since generics are a new formulations, no doubt, but it is a known substance. The other thing where patents can't be given is, say for example, I have a drug which takes care of say for example, some cancer of what do you say, I have a drug which takes care of AIDS, right? And the same drug, if it is used on COVID, the virus, it is the same antiviral drug. There is always a possibility where this drug used for this virus is also effective for this. The same drug, different uses. For different use, you can't have a separate patent granted. When you have a former patent granted, you can't claim the same compound to be acted for another application. So you'll not be granted a patent then. So this is one more thing where patents can't be granted in COVID cases. Fine till here? This one, see, the mere discovery of a new property or new use of a known substance or a mere use of a known process. This simply states that, when aspirin is used, when aspirin was formulated, and when it was found that it can also treat heart ailments, aspirin again can't be granted another patent for separately for treating heart ailments. Fine. Once it is granted, it is done. And people who are using for heart ailments, once the patent expires, once the drug regulatory affairs sit apart, then anybody can manufacture it for heart ailments. Likewise for neem. Neem is known for one of its properties. If you exploit the neem's properties and use it for several other applications, you will not be granted, each application can't be granted a new patent. A mere admixture, if you simply for these combinational drugs right now, people aceclofenac and paracetamol, they simply mix and they simply come up with those combinations. The combinational drugs will not be given a patent unless and until, say for example, I'll explain it to you in a simple terms. You have an umbrella, you have a torchlight, right? And if I fuse them two, if I fuse my umbrella stick with my torchlight, now I just use this torchlight to view the objects there, and umbrella to protect myself from rain. And if I just fuse them two, I will not be awarded a patent. But howsoever, if I concise my arrangement, if I can show that this torchlight when attached or fitted to this umbrella, it's consuming the space I have reduced, or some other advancement features that I have brought which are significant in nature, then it will give me a patent. Likewise, in medicaments, you have the process of coating. We coat the drugs, stunts are coated, drugs are being coated on many things, or the phasic what do you say, extended release tablets are there, the process of release, or there are patches where the therapies can be done. So all these things, the drug is known, but the way it crosses the barrier and the way it reaches the region of interest is what will earn a patent on. And even the barriers that you prepare, the modes of drug transport systems, you involve ligands or you involve some salts, and then let the drugs to cross the barriers and reach out to the region of interest. Even those ideas, as soon as those ideas you think that these are feasible, go for a patent and then demonstrate your idea. So all these can be patented, but not a simple mixture can't be patented, right? The other thing is when admixture or like synergistic formulations, however, is an exception to this. This says that a process of producing a substance, there is an admixture. I mix A, I mix B, I mix C. But if this is not synergistic, like how I mix the paracetamol with ibuprofen, or I mix paracetamol with aceclofenac, but if there is no synergism in the formulation, then a mere admixture can't result in awarding a patent. You should have a synergism that comes up that will give a very good use or increase in efficacy, at least, is what is seen here. Like how I said, and agricultural and horticultural methods, cultivation of algae, producing new forms of known plants, or preparation of an improved soil, these according to this can't be patented. Howsoever, for plants, especially if you develop some new strains or new kind of things, you can have a right under plant varieties. That is a separate right, not included in these 6 of them. Likewise, medicinal, the surgical procedures, or the prophylactic or the diagnostic procedures, the way we do, if knee replacement surgery is done, the process of the surgeries and all, you can't, there is our law doesn't permit those therapies to be patented. No single doctor or no single hospital can own or claim for that particular diagnosis or that particular surgical process per se. However, if I'm using to treat cancer, I have an X-ray machine, I have a plasma machine that goes and affects these cancerous cells. I can claim my patent on these devices, but I can't claim the process of treatment. I can claim the devices. Fine. Like cancer removal technology is there, the process of removal, I will expose the patient to this one, I will precisely cut, the process can't be patented. Howsoever, I can claim or I can patent the telesurgical tumor operating machine. Fine. So this is the major thing. Now, blood transfusions, I can't claim the process of transfusion of blood. Howsoever, I can claim the process where I can store my blood, where the packets, for example, the Sri Chitra University, I think, you'll have fantastic presentation even by Sahu sir, where he will, tell you about one of their inventions, which they have commercialized using that bag. The other things, plants or animals as whole can't be patented. But howsoever, when there is a human involvement, genetic, treatment, like the varieties of microorganisms, they can be patented. That is only an exception to this particular thing. And mathematical business methods or algorithms, everybody pertaining to this, what do you say, the computer based inventions, the programming inventions, people, whenever they claim, when it runs on a particular algorithm, it can't be patented. But howsoever, the hardware component of those computer based inventions can be patentable. Say, for example, I have an algorithm which decides how much dose. I have an infusion system which calculates, which has a sensor placed here. It calculates the amount of blood sugar in the blood, and constantly it gives out the medicament. It releases the medicament based on this particular detected glucose levels. So I will set up a computer program that can calculate, and based on this, the infusion pump will inject. The computer program, the programming, the part cannot be patented when it is not for the first time. But howsoever, I can patent my whole infusion system. Fine. Likewise, drug dispensing things or anything which runs on IoT or computer based systems, I can claim it with a hardware, but not the software as such is patentable as per Indian laws. But howsoever, we have few software patents. I do agree. The Google or the IBM in India, or they hold few of the patents. And when it is dependent on case to case basis, and whenever such cases come up, that we can illustrate how it does. The other thing, the drama or musical work or artistic work, these can't be patented. These can only be copyrighted. These can't be patented. And you develop a game, a crossword, or all those games, a learning language, or you develop an that which will teach, all these are not pertaining to a patentable matter. These can't be patented. Might be they will fit into other IPR rights as copyrights or something else, but not as a patent. These are exceptions only for patents. Presentation of information, the visual display of words or the symbols or diagrams and all, these are also not patentable. These can go into trademarks or can go into copyrights as well, but not into patents. The circuits, the integrated circuits, we come up with circuits, all the layering or the topography of devices and all, they are not patentable. However, in India, we have a separate thing, like how we have a sui generis for plant varieties. Likewise, we have for integrated devices or integrated circuits. So semiconductor circuits and devices. So it can be filed under that, but not as a patent. And traditional knowledge, this is one of the utmost importance, that draws its utmost importance to the pharma knowledge. When we have traditional knowledge, the use of turmeric, the use of neem, or there are certain things, the Ayurveda, or the herbs and formulations related to herbs, or the clove, or whatever we use in formulations of those nutraceuticals and things like that. If they are not new, and if they they are being objected by this section, they can't be patentable. So whenever you have nutraceuticals, whenever you have these such formulations, just take advice on how to, develop them. It usually comes as traditional knowledge. When traditional knowledge is something which has come as an inherited knowledge from our ancestors, it can't be patented. That is the reason why the turmeric, which has been patented in the US, has been revoked by Indians. When turmeric was patented in the US, we fought with US system saying that it was our traditional knowledge. We used it since ages, and we have to show them the manuscripts where we used it, where our ancestors used it, and we said that it was an inherited knowledge for us, and so we revoked that patent. So few of the things like extraction of azadirachtin from neem, this can be patented. However, extraction procedures of something from neem can be patented, but using of neem to antiseptic procedures or antifungal, those things can't be patented. Usually these are the general things that comes into picture of a patent. Title, it has an abstract, it has a description, it will have claims. Now claims are the important heart of the patent. These decide the boundaries, as we have the house boundaries when we claim a property of house. Like claims decide the boundaries of any invention. We have drawings as well. One of the example that I have given, and you can open one of the patents. And the main difference between a provisional and complete specification that we discussed during the time of filing, claims will not be there in provisional application. Without claims, we file all of this. In the complete specification, we file along with claims. That's the only difference. There are two things when it comes to filing of any patent: who owns it and who contributes for the invention. So inventorship, the call, usually when you invent something, when the professors or when the associate professors or faculty or the students make any invention by utilizing the resources in the college, they usually are considered to contribute to the inventorship of a patent. So inventor is someone who conceives the subject matter which is claimed in the patent, and he usually remains as an inventor, the one. And they can be many inventors, they can be principal inventor, who conceives the idea and who gives it, and there can be many other inventors who would contribute at least to a minor part of this patent. When the other people who own the patent are called as assignees, actually they are the owners of the patent. And in case of college, nowadays, after this new innovation policy for the startups, it was released in the month of September in 2019 have come up. It states that college as well as the faculty can co-own the patent application. Usually before that, before this policy came into picture, it was only the person who funds it. Usually the college or the person, the company which funds it, if company, it remains the same. This policy does not influence the companies, it only influences the educational institutes as such. So whenever we talk about educational institutes, the policy says that the educational institute plus the inventor have to own the patent. So what powers does the ownership give? When we sell the patent, ownership decides with whom the rights reside, and these are the person who can sell, who can manufacture, who can do anything with the actual patent. So these are called the actual owners. Assignment is what we call assignee, we call it as assignee. So it can be assignee. And whenever you develop something as an employer to any agency, usually when employee develops something, employee is always being bound by the employee agreement or the offer letters that will be given to you, and it states the extent of IP rights that you own when you work with somebody. So please go through those employment like conditions as well before developing any IP, right? Because though you develop, it might be owned or it might be given to that company itself. So these are few aspects. And one more thing, whenever you claim because once we commercialize, as long as you keep it a patent without practicing it, it is safe. But when you want to commercialize, when you reap, when people when it starts getting money on that, when you sell your patent, we also are involved in the commercialization. Actually, we do sell patents as well to other companies, and we work with the central government, where we one of the institutes called as Andhra MedTech Zone, where we sell patents to different inventors, to different firms which can manufacture the invented patents. So what we do is, when the company gives us the money, we give it back to the inventors, and it depends on, if you have the agreements, then it will decide if you have a co-jointly owned. Say for example, there are 5 inventors who have contributed to the invention. So then the percentage of share which goes to the each of the inventor will be described in a policy. So usually we draft the policies as well, but it is always better to have a policy in place before proceeding in case of co-joint applications, or where you have more than one inventor. And that's it for today. If you have any doubts, I would be happy answering them.
Thank you, madam. Thank you for giving a valuable information. You have explained enormously about the importance of patents and what are patentable and what are not patentable. And you also focused about the how to patent for academic institutions also, you have told, and we are very much thankful. And my personal question is, when we are talking about some issues like drug repurposing. So last time also there was an issue, same drug molecule having different therapeutic activities.
Yes, sir.
Like say for example, aspirin. Aspirin is the oldest drug, which is an anti-inflammatory drug. But now its use is, along with an anti-inflammatory drug, we are also using for antiplatelet aggregator inhibitor. So can we patent for this therapeutic action? So why
No.
not?
When there is a section in our patent, like in the presentation that I have shown to you, sir, according to 3E, it says that no drug, like for another use, aspirin has already been granted patent for something else. But what we did grant a patent is aspirin as such. The product is patented for whatsoever the use was. And this patent can't be, once the patent has expired, this comes into the public domain. It is a known art. Aspirin is not a new compound. It is a known compound. Now, when we claim the aspirin for heart ailments, what are we claiming? We are claiming aspirin only again as a patent, right? But aspirin is already known. It is not an invention as such. It is a known compound. It is a discovery. So you're discovering the new property of a known substance. So technically, it does not fit or come into the purview of the patent law to be granted. So it is not granted a patent for that reason.
Thank you, ma'am. And one of our participants,
yes, sir.
Dr. B.L. Patil from Gujarat,
Okay.
is asking one question.
Yes, sir.
That is, what are the important points to be considered while drafting a patent?
Okay. So drafting a patent, this is a huge, ocean kind of thing, sir. For drafting, there are many things that have to be taken care of, because you describe the invention, but the way you describe, you give your technical papers as such, but the way you present your paper is dependent on the, guidelines, those are to be followed. So there is a Indian Patent Act which says these are the components, like this is the way you have to draft a patent. So we follow generally that part. And as I showed you in the last slides, it should have a clear title, it should have an abstract, the drawing should be there, and for drawings also there will be margins, and the line thicknesses also would be there where we use them. So the line thicknesses and all these there are a certain guidelines which will be maintained, and disclaims also. Up till 10 claims it is free in India, so like the filing fees will be only 1600 if it is an inventor, and if it is a firm or if it's an institution, it will be 8000 per patent. And when we file them, usually what we generally do is, it is 10 claims in this fees that they allow. But apart from 10 claims, if we have to describe our invention in a more elaborative way, the government fee will increase. So it depends on how many claims, and it is all prefixed, the rate is prefixed there. So it should have claims, it should describe all its parts. So it's a description parts, but the side heading should be there. for each of thing, and the document should go on as dictated by the law. That's it.
Thank you, ma'am. Thank you once again for coming over here, spending your valuable time with us. And I request other speaker, Dr. M. Mohan Krishna Reddy Garu from IICT, Hyderabad, to take over the session. And thank you, ma'am. I will call you once the session is over, madam. Thank you. Thank you all.
Thank you so much, and all the best, sir. Mohan
Thank
Krishna.
you. Thank you, ma'am.
Thank you, sir. Bye.
Hello.
Good morning, sir. Dr. Mohan Krishna Reddy Garu.
Yeah, good morning. Good morning, sir.
Good morning. On
So I
behalf
think,
of
it
Vigyan
is
Pharmacy
giving
College.
post to disabled attendee screen sharing. Can you enable it? So
What, madam? What, sir?
that is share screen. You are disabled, I think.
1
Post
minute.
disabled attendee screen sharing.
Yeah, 1 minute, sir. 1 minute.
Yeah, please.
Sir Mohan Krishna Reddy Garu, I think you become the co-host now.
Yeah.
Yeah, you can, 1 minute, sir.
Just check. You're able to see my screen? No?
Yes, sir. Yes, sir. Yes, sir. On behalf of Vigyan Pharmacy College, management, principal, staff of Vigyan Pharmacy College, we cordially welcome Dr. M. Mohan Krishna Reddy Garu, Senior Principal Scientist, IICT. And now I request Preeti, Assistant Professor of Vigyan Pharmacy College, to read out Dr. Mohan Krishna Reddy Garu profile.
Thank you, sir. I, Preeti, Assistant Professor, Vigyan Pharmacy College. It's a great honor to read out the profile of Dr. Mohan Krishna Reddy. He completed his Masters in Chemistry from NIT Warangal in 1919. He received his PhD in Biochemistry from Osmania University, Hyderabad in 2005. He joined as a scientist at CSIR IITR Lucknow, India. He received a Boy Scouts Fellowship to pursue his postdoctoral work at Johns Hopkins Medical Institutions, USA in 2007. He authored more than 80 publications in reputed national and international journals with more than 1980 citations with an H index of 27. He successfully completed 6 projects as principal investigator and maintained his laboratory as per ISO 17025 guidelines for analytical testing in pharmaceutical, food and environmental areas. He received the following awards. He awarded 8 PhDs till date and currently guiding 10 PhD scholars. He is an acting academic editor to PLOS ONE journal and also serving in various national committees. He visited countries like Australia, China, France, Germany, Taiwan, and USA as a part of his scientific collaborations and assignments. He is a recipient of ABAP Young and Senior Scientist Awards. With this, I welcome Dr. Mohan Krishna Reddy Garu to start the session. Thank you, sir.
Yeah, thank you, Preeti, and thank you, Vishwanath Garu, for a nice introduction. And I hope I am audible to everybody. I'm clear to you all.
Yes, you are audible, sir.
Thank you. So today my talk is on illustrating about what is mass spectrometry and how it is helpful to the pharmaceutical research. So that is my talk, and I belongs to CSIR Indian Institute of Chemical Technology, which is a 75 years old institute constituted under the aegis of Council for Scientific and Industrial Research. So our department in IICT has 4 research groups. One is working on NMR. It's a world-class NMR facility at our institute, having 9 NMRs in our institute now. And then mass spectrometry, then chromatography separations and trace elemental impurities and thermal analysis. And also we have a exclusive X-ray crystallography unit to support the mainly for pharma and biotech industries, apart from food and environmental research. So when it comes to our institute capabilities with respect to the instruments what we have, So we have a wide range of instruments starting from HPLC to almost everything. I think I can say with a 95% confidence, I can say that you name the instrument and we have that. So we have instruments for all type of analysis requiring from organic analysis, either organic and many of them, and inorganic analysis, and even thermal and structural characterizations, and also crystallography. So we have, you can see more of the details about the instrument in our website. And also we have been accredited with NABL and as per ISO 17025 standard guidelines in the areas of active pharmaceutical ingredients and oils and fats and water testing and warfare chemicals testing. And also we have been now in the process of getting GLB certification to our analytical facility in the areas of physicochemical testing and pharmaceuticals characterization and analysis and biosimilars characterization.
Sir, sorry to disturb you, sir. Mohan garu. Sir, screen share cheyala, sir.
Screen, I have already shared it, na?
Rale, sir.
1 minute.
We have given you the option, but ఇందాక వచ్చింది సార్ మళ్ళీ కట్టే ఐ థింక్ ఎస్ సార్ నౌ యూ కెన్ కీప్ ఇట్ యాజ్ ఎస్ సార్ వెరీ గుడ్
Now it is coming, na?
ఎస్ సార్ అండ్ ఇఫ్ యూ హ్యావ్ ఇయర్ ఫోన్స్ ఇట్స్ బెటర్ టు యూజ్ ఇయర్ ఫోన్స్ సార్ దేర్ విల్ బి నో డిస్టర్బెన్స్ ఫ్రమ్ అదర్ సైడ్ థ్యాంక్ యూ సార్
Now it is okay. Any voice disturbance you are observing? Because I'm in
it's okay,
the room
sir.
to
It's
close
okay.
the environment. Nobody is there in my room at all. Huh?
లిటిల్ లౌడర్ వాయిస్ ఈజ్ ప్రిఫరబుల్ సార్
Okay.
థ్యాంక్ యూ సార్ థ్యాంక్ యూ
Right. So in the recent times, we have been our facility. We have been providing a lot of analytical services, to the pharmaceutical industry, for which many times when the pharmaceutical industry will go to the FDA filing to market their products in USA. So at that time, the FDA will inspect our facility and see that whether we have been meeting with the FDA norms as per the pharmaceutical characterizations. So we have been inspected 3 times by the Food and Drug Administration of USA, and this time we have been qualified the FDA inspection without any observations or any non-conformances. So that is one of the greatest achievement from our institute, that we have been awarded a no establishment inspection report, that is as no action initiated. So that means there is no 483 document been issued. So this is our jewel of the crown that shows that the confidence on our analytical facility by the international regulatory communities. So we have been continuously providing services, analytical services, as well also providing the customized solutions as per the client requirement. So when it comes to the services, we have been routinely performing the structural characterizations by NMR, mass spectrometry, and some spectroscopic techniques, and also purity assays by liquid chromatography, then genotoxic impurity analysis using GCMS and LCMS, and trace elemental impurities in active pharmaceutical ingredients by ICP OES, and chiral analysis by liquid chromatography, and LC-based purifications. We have preparatory and semi-prep instruments with us. And also we have been continuously carrying out the quantitative analysis as per the requirement, and particle size and surface analysis, and drug polymorphism, as well as crystalline phase, even the single crystal studies also we can perform with our facility. And also the solutions will always be need-based, depending on the type of the solution, what the client wants, so that we can customize and we can provide that, because that a lot of infrastructure is available with us with respect to analytical method development and validation, impurity profiling, pharmacokinetic studies, and biosimilars characterization, untargeted contaminants analysis, and stability studies for active pharmaceutical ingredients, including force degradation studies, as well as metabolomics, and also performing skill development programs. The biosimilars characterization, which is a now emerging market in India also, because vast expiry of the patents related to the innovator biologics molecules, which has been now people have been trying to produce the molecules similar to the innovator reference, that is called biosimilars. So to establish a compound or molecule as a biosimilar, we need to perform a lot of analytical characterizations as per ICH Q6 guidelines, Q6 document guidelines. So including that includes physiochemical, structural characterization, confirmation, and impurities, contaminants, and degradation products. So we have been received a BIRAC grant to establish a GLP compliant analytical facility for this biosimilars characterization. So that project is under progress now. And we have been routinely performing the skill development programs. Nowadays, there will be a lot of emphasis on the skill so that the student who finished his post-graduation, then after the skill development course, he can, from day one onwards, he can work, ready to work at the industry. So that is our motto. And we generally design the skill development program based on the need of the student group or the employee, because sometimes the employees will come, they may not require to have a basics of the theoretical aspects of the basics of the instrumentation. So they need some advanced training. So as per the need, we usually provide the training. So far now we have been conducting skill development programs for postgraduate students who just finished their MSc or M.Pharma. They can come and have a skill development training at our institute for 4 to 6 weeks. And then we have regulatory officials like Excise Department and Central Revenues Control Laboratory. So this CRCL training we have been conducting 30 batches. So far we have finished 5 batches. And we also been awarded one international skill development program from OPCW, Organization for Prohibition of Chemical Weapons, which is a GCMS based training basically for 2 weeks from the participants from 20 different countries. So we have finished 2 trainings of that sort, and next 3 more we have to conduct. So 5 years we have been awarded to conduct that training program. So that's all the whole about our institute activities with respect to the analytical perspective. So the mass spectrometry, the today's topic, is a tool which is emerging, is a established and nowadays advanced tool to measure the molecular atomic weight. So this is very important tool, and because of its advantages with respect to sensitivity, selectivity, and also hyphenation to the separation techniques like liquid chromatography and gas chromatography. So that is one of the most important aspect. The hyphenation has expanded the scope of the MS to be used in the pharmaceutical and biotech industry with respect to identification, characterization, and also quantification. So there are many types we have been used. Now mass spectrometry can be used. Mass spectrometry basically is an instrumental technique in which sample is converted into rapidly moving positive ions, which are then separated and characterized. The advantage of mass spectrometry is it can provide the information and also fragmentation of the compound, and also since it is a highly sensitive technique than any other spectroscopy techniques. If you compare the sensitivity of mass spectrometry with NMR is a good technique for organic chemists. But when it comes to the quantitative analysis or the impurity profiling of the active pharmaceutical ingredients, so that time the mass plays a vital role because of its high sensitivity, and it is capable of high precision and accuracy during the analysis and provide measure of the molecular weights. So when it comes to the history of the mass spectrometry, it is first been prototype mass spectrometer was being designed by J.J. Thomson, and who have been awarded Nobel Prize in 1906. And then mass concept first put into practice by Francis Aston, the physicist working in Cambridge, and he has also been awarded a Nobel Prize. And this mass spectrometer generally designed to measure the mass of the element. And then after that, there are a lot of advancements been happened in mass spectrometer, designing the different type of mass analyzers in mass spectrometry. But the first one is time of flight, then quadrupole, then tandem mass spectrometers, that is coupling of one mass spectrometer with another mass spectrometer in the same instrument. And mass spectrometers are now one of the most powerful analytical tools in chemistry. So the type of information generally the mass spec gives is, one is molecular weight. The second one is elemental composition. When you use high resolution mass spectrometers, we can even get what are the type of the elements present in our compound. And also it can provide the structural information that depends on the type of the ionization as well as the collision induced dissociation we are been using. So when it comes to the mass spectrometry, for small organic molecules, the molecular weight can be determined within 5 ppm accuracy, which is sufficiently accurate to confirm the molecular formula from mass alone. That means we are not using any hyphenation, that is any chromatography technique. Simply directly we are injecting the sample into the mass spectrometer, which can give the structural identity of the molecule with an accuracy of 5 ppm. So that is even for the now high resolution mass spectrometers can able to predict the structure within a mass accuracy of 5 ppm. For larger biomolecules like proteins and carbohydrates and nucleotides, and basically the molecular weight can be determined within an accuracy of 0.01%. That is within 5 Dalton for a 550 kilodalton protein. So generally 1 Dalton is assumed to be 1 atomic mass unit in the mass spectrum. So if you see any element in a periodic table has its mass, one is atomic number and then is a mass, atomic weight of the element. So based upon that, each compound will have a identity depend by the mass of that compound. Suppose if you see L-DOPA, which is having a molecular weight of 197.2, that is a uniqueness for the structure for the compound, which is been used to analyze this compound by mass spectrometer. And like ethanol, it is 46.1. So when you see the mass spectrometer, how it will works and how it able to accurately determine the molecular weight of the compound. So that the mass spectrometer generally contains 3 components. One is the ionization of the compound, for which we need ionization sources like MALDI, that is matrix assisted laser dissolution ionization, and then electrospray and many other. And also then after that, once ionization has happened, if many of the ions have been or fragmentation have been happened, how to separate those fragmented ions, that for which people generally have the mass analyzer, that is that will separate the ions based on the mass by charge. And then each separated ion will go to the detector. That detector will be based the photo multiplier tube or a electron multiplier tube. So that will detect the ions when reaching to the detector based upon the current produced by the ion. So that's how the mass spectrometer it will works. So it is having, one is ionization, the second one is an analyzer, the third one is detection. This is all three is a combination which is called as a mass spectrometer. So when you see the mass spectrometer principles, one is sample when it is introduced into the ionization chamber in the mass spectrometer, where the sample will be ionized. Ionized means that is a neutral molecule which has been bombarded with an electron or some photon energy or some spraying energy, voltage, basically based on voltage. Then the molecule will be ionized. The ionized molecules will go to the analyzer, at which the ionized molecules will be separated based on the mass by charge. Then each separated mass will go to the detector, at which based upon the intensity, the number of ions reached to the detector of the same molecular weight, the peak intensity will be observed. So that is the whole principle of the mass spectrometer. So when it comes to the in detail, what are the ionization methods basically used in mass spectrometer? One is electron impact ionization. That is a hard ionization method. The high energy electrons will bombard onto the molecule, so it will give the fragments. The second one is chemical ionization. So the chemical ionization is something like we call it as a soft ionization, which has been used to understand the molecular weight. The third one is electrospray ionization, which has been generally used. The ions in a liquid form will be sprayed like a gaseous molecule. So that is a electrospray ionization. And then atmospheric pressure chemical ionization. Sorry. Atmospheric pressure chemical ionization. Then fast atom bombardment. Then field distortion, field ionization. Then matrix assisted laser distortion ionization and thermospray ionization. So many of the techniques people have been using, and fast atom nowadays it is becoming a too conventional, and field distortion also. So most widely used is electron impact and chemical ionization, which has been immensely helpful when the mass is coupled with gas chromatography. So if you have electrospray ionization, then atmospheric pressure chemical ionization, which have been most helpful when we couple mass spectrometer with a liquid chromatography. So if you have a MALDI, a matrix assisted laser distortion, which has been used for the biomolecules molecular weight determination. So if you can couple with the time of flight mass analyzer, it can be MALDI-TOF. So when it comes to the electron impact ionization, which is basically the sample, the compound will enter into a mass spectrometer. So at which we have a filament. So when we heat the filament, it emits the electrons. The electrons will, this is the filament. So this filament will, the sample is coming from here. This is the sample inlet. So the samples are coming, the filament will emit the electrons, electrons will bombard with the molecule. So then this molecule will be ionized and also simultaneously fragmented, because the energy of the electrons what we are using, generally it is a fixed 70 electron volt. So at that energy, this molecule will be fragmented. So all the fragmented ions will go to the mass analyzer. So we have a repeller here, then we have a other aspect, so that we can focus the ions to a mass analyzer path. So this is the electron ionization. So sample introduced in instrument by heating it until it evaporates. Gas phase sample is bombarded with electrons and molecule is shattered into fragments. So here simultaneously ionization as well as the fragmentation of the compound has been happening. And the fragments then sent to the mass analyzer. The mass analyzer could be a quadrupole, could be a time of flight, could be Orbitrap, could be ion trap. So when we see some simple molecule methanol, so when it goes to the enter into the EI source, so where the immediately it will be ionized, ionized one will be bombarded with electron, and there is a lot of fragments will be formed, like CH2 double bond H. This is the one aspect, and there is also chance of bombardment can result to methane ion as well as hydroxy. Then there is a lot of fragments will be formed. So once this fragment, the mass spectra will be acquired by the detector, the masses ions after analyzer, and it will go to the detector. So they will be recorded. So if you see 16, methane is the 16 molecular weight, it will give a 1 peak. Then there is other fragment is 15. So that may be CH3O, like that. H proton is released from the OH. So if you see here, this is the molecular ion, the molecular weight of the molecule, generally we termed it as molecular ion. And the base peak in the spectra, out of which suppose you have 2 ions here, the most intense ion, that means it has 100% relative abundance, that is a 16 is having a most intense, so that also called as a base peak. So this base peak generally used as a qualifier ion for the quantification of methanol by using a mass spectrometer. So what type does a molecule look like? Typical mass spectrum. Suppose if you see a mass spectrometer, suppose the compound with a molecular formula of 114, the mass by energy will be in x-axis and y-axis will be relative abundance of the ions. If you see here, this 43 will be the base peak because it has a 100% relative abundance in the whole fragments of 114. So this will give a mass spectra, how it will looks like once it goes to the fun. So base peak given 100% abundance will be 43, and then the molecular ion will be, generally we assume that the molecular weight of the compound will be the molecular ion. So then there will be, suppose if you see here, there are some small ions also which are close to the ion, so that could be resulting due to the isotopes of the element. Suppose if you assume that carbon exists in C12, but sometimes C13 some percent will exist. So that will show as a C13 means 43 plus 1, so something 44. So like that, this ion isotopes will present in the bunch as a bunch. So if you see here fragment, this is the molecular ion 114. Then we can see the 71 is there. The difference is 43. So 43 is the fragment due to the the conversion of 114 to 71. So that's how we can interpret the mass spectrum in our structure. Sometimes when we see that, if you see this is the molecule amphetamine. Amphetamine has a molecular weight of 135. But if you see the spectra, we don't have 135 here. There is no molecular ion. So this electron ionization is a fast bombardment, basically high with high electron energy, high energy of the electron. So sometimes we may not see the molecular ion of the compound. So then the fragmentation pattern will be unique for that. So we are fixing the electron energy at 70. So due to which the fragmentation is always reproducible. So here, if you see the spectra, the 44 is the base peak. So if you just check with that compound, what could be the structure? So that's how we can just see the intensity. So many times in EI spectra, EI mass spectra, we may not observe for molecular ion because of high bombardment, due to which the molecular ion may not be stable and immediately convert into the fragments. if you see a mass spectra of the morphine, so if you see this is the molecular weight of 285. 285, you see the, and this is the molecular ion as well as the base peak of this morphine. So there are other fragments. So when you run this morphine in EI, electron ionization, even 100 times, even 100 days, you will get the same fragmentation. So due to which we can able to produce a libraries based on the EI electron ionization source. So that's the reason the GCMS, the gas chromatography coupled with mass spectrometer, will always have a
బోర్డ్ లో చేస్తే నాకేమో అది ప్రాబ్లం అవుతుంది. పద్దానికి 100 కేజీలు చదువుతాను.
తప్పదు ఇంకా మనం మొత్తం ఏం చేస్తాం, ఇంకా వాళ్ళు చేసిన మన తప్పు కాదు కదా, అయినా మనం ఏదో కష్టపడాలి ఇక. రండి ఇక, సరే అది కాపీ చేసుకొస్తాం మరి పెన్ డ్రైవ్.
So shall I go ahead? So this is how this is. That's why the EI electron ionization will always have a libraries. That's the reason GCMS will always have a lot of NIST library, National Institute for Standards and Technology mass spectral library, which is almost 2 lakhs compounds have a reference spectra at which we can identify the compound by easily by the spectra getting from the instrument with the comparing with the library. Then the second one is chemical ionization, which is basically a soft ionization process, we call it as. This is a gas phase chemical production of ions in the source chamber of mass spectrometer, which is proposed by JMS Hennis. And with EI, MS spectra of anactodiene contains the fragments with 4, 5, and 6 carbon atoms were obtained. No molecular ion was appeared. Suppose we have seen in that earlier chromogram also, there is one compound amphetamine which doesn't have molecular ion. So sometimes if the spectra is not available in the library, it is very difficult to interpret what could be the structure of that compound. So for which we have to understand what is the molecular weight. So that's the reason people will always use chemical ionization to identify the molecular ion of the compound in the mass spectrum. So that is the reason we always use chemical ionization, because it is a soft ionization. So how the software we use some gases like methane, ammonia, and also some ammonia and isobutane also people use. So what happens here, the electron emitted from the filament will not directly hit the molecule. So it will initially the electron will bombard with the gas what we are using. Suppose we are using a methane gas, so electron will ionize the methane gas. That ionized methane gas will bombard with our molecules. So this is our molecule. So this ionized methane gas will bombard. So that means the electron directly not hitting the molecule. So due to which the ionized methane gas hitting, so the ionization efficiency or the bombardment with our molecule is very low. Due to which we definitely get the molecular ion in the mass spectra. That's the reason the chemical ionization will always be used to determine the molecular ion of the compound. Then comes to the other ionization sources like electrospray ionization and atmospheric pressure chemical ionization. These are the two modes we generally use it for when we couple the mass spectrometer with liquid chromatography. So here the ionization is once the liquid, that is liquid chromatography means once the mobile phase or compound we are injecting in the liquid, and that compound will before entering into the mass spectrometer will comes to a interface. So mass spectrometer will always operates in a vacuum. And LC generally it is a atmospheric pressure. Why mass spectrometer always operates in a vacuum is because the ions after fragmentation has to move from ionization to mass analyzer, then to the detector. So the movement of the ions should not be restricted by the presence of any air molecules or any other gas molecules. So due to which, to avoid any hindrances, because if any hindrance is there, the movement will be restricted. The ions may not reach to the detector at a regular interval. So this may hamper the reproducibility of the fragmentation occurring in the mass spectrometer. So for which, that's the reason we always operate mass spectrometer in a vacuum mode. So but LC is in atmospheric pressure. So what could be how this liquid from the liquid chromatography will go to the mass spectrometer, which is operating in a vacuum. So for which there is a interface, there is a term called interface, which has been designed so that the molecule will not directly enter into a liquid as a liquid into the mass spectrometer, because if liquid enters in mass spectrometer, it definitely affect the vacuum status of the mass spectrometer. So that's the reason the ionization sources are designed in such a way that the molecules in the liquid will be enter as a spray. The spray means that is something like aerosol droplets which has been spraying. Something like you can see the deodorant. If you press the deodorant, you can see the gaseous molecule. It is a liquid, although it is liquid, pressurized gas. It is a liquid type, and how the droplets will be coming from that deodorant spray, like that only the spray will be happen. So we have been given some voltage so that the desolvation will occur once during the spray process. The desolvation means the solution, the solvent used in the liquid chromatography will be evaporated. Then the ions, the molecule, only the compound ions will be ionized, and those will be sent it to the mass analyzer for separation of the ions, as well as then to the detector to detect the ions. So that is the electrospray ionization. And then comes to the other one is atmospheric pressure chemical ionization. This is nothing but a chemical ionization, and it is similar to the electrospray ionization. But only difference is that here we are being directly giving voltage to our molecule, and we are doing the desolvation process. But here the outlet of the liquid chromatography will go to there is a needle called corona discharge needle. So where we have been giving a voltage to that needle, so that will give slowly, it will give the energy to our compound. So that's the reason the ionization will occur in atmospheric pressure using a corona needle. So this will ionize the molecules. The ionized molecules then will go to the mass spectrometer. The difference between electron ionization and electrospray ionization is electron ionization, we will get the total fragmentation in a single attack, single bombardment. But here, in electrospray ionization, initially the molecule will be ionized. So if you want to do the fragmentation, you have to perform then MS-MS. That's the reason the tandem MS, that MS coupled to another MS, is gained prominence due to the design of electrospray ionization as well as APCA. And many of the polar metabolites can easily be polar compounds. That means compounds with polar functional group like hydroxy, carboxylic acid, amides and all can easily be analyzed with LC coupled with mass spectrometer. So LC MS-MS, that's the reason it has gained prominence in recent times in pharma and biotech industry. When it comes to, I'm taking only 4, and many other them you can read it. If you have any queries, you can just mail to me. And if you see the summary of the ionization techniques, electron ionization, these fragments means molecular ion is the molecular weight of the compound. Here we will get the M+. The M+ means if the compound has 16 is a molecular weight, it will show 16. So like that. So if you see electrospray ionization, that is in 2 forms. One is positive electrospray ionization and negative electrospray ionization. So it will have ionization and will take a proton adduct. So that is hydrogen. So it always gives the molecular ion is molecular weight +1. Sometimes there is a possibility of adduct formation in this, that is like M+ ammonia, M+ sodium. So when we see your molecular weight is 23 and you get a molecular ion of 39. So that means, 16 is the molecular weight and you got a molecular ion of 39. That means you assume that your compound M plus sodium, sodium is 23. So that adduct formation is quite possible in LC MS-MS. So when we see that, that you may not be wrongly erected, you have that adduct formation will mainly depend on the type of the mobile phase or the solvent we have been using for dissolving the compound. So based upon that, we can get. And there is a negative mode also for it. Many of the compounds which have electronegative groups, so at which, so they have electron attracting tendency. So they can give a M-1 or M+, molecular ion and molecular ion -1. So that type of can give. And even APC also can give M+H and M-H and rest of. There is another atmospheric pressure photoionization. This is mainly used if you are using LC-MS for non-volatile compounds like hexane, benzene. These are all said to be non-volatile. These are volatile but non-polar. So for which we have to use this atmospheric pressure photoionization. Here, instead of corona discharge, we are putting a photoionization source, that is UV lamp. So that it has krypton, which will emit photons, and photons will bombard with the molecule and then ionize the molecule. So that is basically the ionization mode will depending on the type of. When you are using a voltage directly to the molecule, that is electrospray ionization. if you are using a corona needle, and that means the indirectly we are supplying voltage through corona needle to the ion, that is atmosphere pressure chemical ionization, APCA, and we are using a photon to bombard the molecule, that is APPA. Likewise, matrix assisted laser desorption, in which we are been passing a laser onto the directly to the compound before, and there is some matrix. The matrix will absorb the laser lamp and supply the energy to the molecule for ionization. So at which we will get the molecular ion plus hydrogen, and also some adducts can be possible. So if you see the application range of ionization techniques, many of the compounds like sugar, small ions, amino acids, organic acids, flavonoids, peptides, and some of the lipids and the liposoluble vitamins and steroids can be easily be analyzed by electrospray ionization. And some of the non-polar compounds or semi-polar compounds can be easily analyzed by APCA. And non-polar means like hexane, benzene type of compounds can be easily been analyzed by APPA. So, but APPA is a little bit expensive technique to use, and many times if you are using APPA means instead of that, if you have a GCMS, which can solve the purpose. So if you have GCMS and LCMS, that's the reason the techniques are always complementary to each other. The advantages and disadvantages of these ion sources have been plenty. So based upon the need, because every ionization has its pros and cons. So the advantage of ESI is it is applicable to wide range of analytes, and the thermal levelity and volatility usually not a problem, and minimal fragmentation of molecular ion, because here initially if you are using only single mass spectrometer with ESI, that means you will get only molecular ion. So if you want to do fragment that molecular ion, you have to do MS. And higher molecular weights can be easily analyzed, and high sensitivity can be achieved with this ionization source. The disadvantage is ionization suppression that could be possibly due to the use of the mobile phase or flow rate, or the energy required to ionize the molecule. So that we have to optimize during the method development aspect. And mobile phase composition can also affect the sensitivity of the analysis. So APCA and APPI, it has low polarity molecules can easily be ionized, and it has wide dynamic range and mass sensitive, and higher buffer concentrations. Even then also the ionization efficiency will not be affected if you are using APCA and APPA. And the disadvantages is analytes must be thermally stable. That is one of the most important thing. And analytes must be volatile. And if you are needing a dopant, sometimes the ionization efficiency of the APPA, the photons, may not be sufficient enough to ionize the molecule, for which we have to use some dopants. So that's the reason this is little costly an ionization process in LC-MS analysis. So once we are ionizing the molecule, then separation has to be conducted. The separation is only the cations are deflected by the magnetic field. Amount of deflection, the ion deflection depends on the mass by charge. The detector signal is always proportional to the number of ions hitting it. By varying the magnetic field, ions of all masses are collected and counted. So the initial times when the mass spectrometer was designed, the magnetic sector mass spectrometers, which can give a high resolution mass of the compound. But since nowadays, because of the length of the instruments and the cost involved in it, and sometimes the sensitivity associated with the analysis has now reduced the use of the magnetic sector mass spectrometers. But still people are using for the compounds like dioxins, furans. Suppose if you assume dioxin, one compound will have 16 congeners. So they are very closely associated. The mass of the 16 congeners are same. But if you see the accurate mass will be little different. To separate them, still people are using magnetic sector based mass spectrometers. And the deflection, this all ion focusing and the process, ionization process, and then ion focusing will always to improve the deflection, ion deflection at the magnet. So if you see here the magnet, here the deflection will be always gives the accuracy and sensitivity of the analysis. So mass analyzers, what we have is low resolution. Low resolution means the analyzers would can able to discriminate the ions with a mass unit of 1. That means if we are analyzing with a quadrupole, if it is having a molecular weight of 214, then it can only discriminate 215 and 214, but it will not do 214.22. If it is there, it will not able to identify. It will show it as a 214 only. So that is called low resolution mass spectrometers, and even ion trap. And people are now the high resolution is time of flight. So that is the mass analyzer, which it can even discriminate at the digits after decimal. The sector magnetic sector instruments and ultra high resolution is isotron ion cyclotron resonance. This is also a very high resolution, but its usage is very limited to some areas. So when it comes to the time of flight, it is the measure of the time required for the ions to fly down to a length which can reach to the detector, from ionization to the detector. That is the mass analyzer, TOF. So based upon the movement of the ions, the velocity of the ions, we can able to determine the mass of the compound. And a tandem MSMS, which can use not only separation, but also identification and even for the quantification, which can induce the fragmentation and mass analyze the fragment ions. So the tandem MS can be with the different combinations like quadrupole, that is low mass resolution, but it is very good technique for to get the reproducibility with respect to the quantity of the analyte in the sample. And then magnetic sector quadrupole, this is a high resolution mass spectrometer. One is magnetic sector, which is HRMS, and quadrupole is LRMS. Both have been hyphenated to give reproducibility in the quantification, but also accuracy in the mass measurement. And then quadrupole time of flight, which has been very widely used nowadays for the biosimilars characterization, like complex molecules like proteins and peptides and carbohydrates, even polymers analysis. The quadrupole time of flight and time of flight. So there is a possibility of different combinations we can able to design in the tandem mass spectrometers, and then we can hyphenate to the chromatography techniques like liquid chromatography and gas chromatography. So if you see the mass accuracy and then resolution for the perspective of each mass analyzer, the quadrupole has a mass resolution of 2000 resolution with the accuracy of 100 ppm. Then TOF, if you can see the TOF, which is having a mass resolution 10,000, but it is going up to 70,000 almost nowadays with advancement technology. This is a little old slide and Orbitrap can give a mass resolution at 1 lakh with a mass accuracy less than 5 ppm. This is very much that means if it is the mass accuracy or mass error is less than 5 ppm, that means the prediction of the measure of the mass of the compound by Orbitrap is very highly accurate. And FTICR is a very high 10 power 5 of the resolution with a less than 5 ppm mass accuracy. So then after the mass analyzer, then we have to, once the ions are separated by the mass analyzer, then it goes to the detector. The type of detectors generally, mass detector will detect the current signal generated from the ions after separation from the mass analyzer. Different mass spectrometers are used to detect depending on the type of the mass spectrometer. So most commonly used is electron multiplier, Faraday cup, photomultiplier, high energy diode, dynode detector, array detector, microchannel plates, and charge. Most widely used is photomultiplier tube, and then electron multiplier tubes. So these are the most widely used in the LCMS or GCMSs. So when it comes to the mass spectrometer, the isotopic abundance also will play a vital role in predicting the structure of the compound. If you see hydrogen, which is having a 1H is 100%. There is no deuterium or tritium existence because this is existing almost 100%. And carbon 98.9 and 13C will be 1.5. So there is a chance of 13C, one ion will be C if the compound contains carbon. The nitrogen 99.6 and 0.4% of the 50 nitrogen. Oxygen 16 oxygen is 99.8 and this is very less. It may not be reflected. But if you see the chlorine, 35 chlorine, it is exists in two isotopes. One is 35 chlorine and it is 37. So 35 chlorine exists in 75.5% and 37 chlorine exists in 24. That means 75 is to 25. And bromine exists in two isotopic forms. One is 79 bromine, that is exists in 50.5% in the nature, and 81 bromine is 49.5%. So that means this is 1:1. And iodine is no only single isotope, that is 127, that is 100% is existing. So depending on the isotopes present in the to each element, the isotopic abundance will be observed in the mass. If you see here is a ion, we are one ion, but the due to isotopic abundance, there is M+1, M+2 like that. So this is the molecule which is having a molecular weight of 76. So here we have a sulfur. If you see this here, sulfur is sulfur is 32 sulfur 95%, and 34 sulfur will be 4.2. So that means there is a one isotope, 34 isotope also can coexist here. So if you see the molecular ion, the 76, and it is having M plus to 78 in a ratio, we can see the intensity ratio, it is almost 95 to 5%. So you can say that the compound contains sulfur. So like that we can interpret the whether any halogen atoms or any other except the carbon hydrogen, any other atoms are present in the structure, which can give a clue for this. Likewise, chlorine. If the compound contains chlorine, the chlorine exists in two isotopes. One is 35 chlorine and one 37 in 3:1. So if you see here, this is the 78 molecular weight for this compound, and then there is a one more ion, which is saying 80 molecular weight. The ratio, if you see, 75:25. That means 3:1. We can see by seeing this spectral pattern, we can say that the structure contains chlorine. Likewise, it will helpful when you identifying a unknown compound by using mass spectrometer. Likewise, if you see, this is the compound which contains a bromine. Bromine exists in 79 bromine and 81 bromine, 1:1 ratio. So here, if you see molecular ion 122, and then equally there is one more ion which is having a M plus 2. That means 81 bromine is been added. So if you see this spectra, you can say that the structure contains bromine. So it will give a clue when we are identifying the structure of the compound. So the molecules with the heteroatom isotopes present in their usual abundances, that is nature abundances. Hydrocarbons contain 1.1% of C30. So there will be a small M plus 1 peak. If bromine is present, M plus 2 is equal to M plus. If chlorine is present, M plus 2 is 1/3 of M plus. So if iodine present, there is no other isotope except 127. So there is only single ion will be shown. If nitrogen is present, N plus will be an odd number. So that means this is called a nitrogen rule in mass spectrometer, at which we can say if molecular weight is odd, then we can have an odd number of nitrogens present in the structure in the compound. If sulfur is present, M plus 2 will be 4% of the molecular ion. So this is how we can able to interpret the mass spectra for your compound. So then there is a tandem mass spectrometer, which has been gained also very prominence because of the type of the ionization what we have been using. So the tandem mass spectrometer, the purpose is to fragment ions from parent ion to provide structural information, and also allows mass separation and identification of compounds in complex mixers. Because nowadays active pharmaceutical ingredient, the matrix will be interference will be very low, because that is almost a purified compound what when done synthesized by the organic chemist. But when we are identifying the same compound during the pharmacokinetic studies or toxicokinetic studies, the sample what is basically serum, urine, plasma we are taking. So that is very complex because it contains not only proteins, carbohydrates, or fatty acids like many other things. So we have to eliminate the matrix interference, for which we need a tandem mass spectrometer, because it can allow mass separation and identification in complex mixes like when we are dealing the bioanalytical work. And uses two or more mass analyzers filter separated by collision cell filled with argon or xenon. And collision cell is where selected ions are sent for the fragmentation. This is a tandem mass spectrometer. So if you see the advantage of tandem mass spectrometer is in the first mass spectrometer, if you see this figure, so if you are using only single MS with ESI ionization, we can get the single MS, we will get the molecular ions. Then if you want to understand the fragment of this, then you have to MS. So for which we have to attach one MS with another MS. Then only we can able to see the MS scan of the molecular ion. So, but sometimes this is the advantage. If you have a single mass, then you have to perform the sample in 2 times. One is initially the mass, we can get molecular ion, then you have to perform the fragmentation study in second run. That means 2 times you have to run single sample. But you have a tandem MS. That means tandem is MS is hyphenated to another MS. So it can simultaneously give the molecular ion as well as the fragmented ions. So that's the reason the tandem mass spectrometer has gained prominence for in pharmaceutical analysis. Generally, how this MS setup will be, sample will be introduced into the ionization source. Suppose if you are assuming EA is the ionization, the fragmentation will occur at the source itself. Ionization as well as fragmentation at the source itself, it will occur. Then the fragmented ions will be separated by first mass, MS1. After that, we want to see, if you want to see out of the fragmented ions, I want to see that this ion, suppose it is a purple color, this ion, what is the fragments for this ion? Then I have to perform a MS, tandem MS. In tandem MS, we have MS1, MS2, and MS3. But this here MS2 will not act as a mass analyzer, but it will act as a collision cell. So this collision cell, after that, after the one mass analyzer separating this ion, we are selecting this ion, we are sending to collision cell, at which we are giving organic gas here. The organic gas will bombard with ion, will get the fragments, and these fragments will send to the mass analyzer, that is MS2. So this is called tandem mass spectrometer. Then all the separated fragments will go to the detector to detect the ions. So this MS tandem MS, that's why MS means two mass analyzers. Although there are three mass analyzers are there, but the second mass will be used as a collision cell. So it will only fragment it. It will not analyze the ions. So the third MS, that is MS2, will analyze the fragmented ions from the collision cell. So that's how this MS will work. So in low resolution, high resolution, and there is a lot of difference between these two mass spectrometers. The high resolution mass spectrometer, which can measure the mass at to 1 part in 20,000. That means the example is, suppose you have a molecular weight of compound 44. So if you see these formulas, this is C3H8, C2H4O, CO2, CN2H4. All this will give a 44. This is a trivial molecular weight. But if you calculate the exact molecular weight of this compound, it will be 44.029. So now what I did, I analyzed this compound by using high resolution mass spectrometer, which is given 44.029. But if I calculated the exact mass of these compounds, C3H8 will be 44.0626, and this is 44.02620, and 43.98, 44.037. All these will be 44. But if you see, the instrument has give 44.029. So that is closer to 44.026, C2H4O. So that's how the high resolution will help you to analyze the compounds with same molecular weight. So where we can get the high mass accuracy to predict the structure of the mass of the compound. So the high versus low resolution ESI is QTOF and ICR will be high resolution masses, and ion trap and quadrupole will be unit resolution. Unit resolution means 44 and 45 will be discriminated in the mass spectrometer, but 44.02, 44.03 will not be discriminated by using quadrupole mass spectrometers. So mass resolution is one of the important factors when we are using high resolution masses. The mass resolution, it is the ability of the mass spectrometer to separate two adjacent ions of masses M1 and M2 differing slightly in M/Z ratio. So this is the how we will calculate the resolution of the mass spectrometer required to separate the closely associated mass spectrometers. So here, if you see R is equal, that is the resolution, is equal to molecular weight mass by difference in the masses of the two ions. That is delta M is equal to M1 minus M2. So at peak at half height, if we take, we can calculate the resolution required to separate the ions. The example is, suppose two compounds which is having a nominal mass of 321, both having nominal mass, but can we differentiate these two ions based upon high resolution mass spectrometer? But the exact molecular weight of the each element will be like this. And when we calculated the molecular weight of this C24H19N, it will give an accurate mass of 321.1513. And the other one is C21H23NS, which is having an accurate mass of 321.1546. If you see here, they are very closely associated. Then how we can able to discriminate or separate these masses by using mass spectrometer, and what is the resolution required to separate these two ions? So that when we calculated the resolution M by difference in the two masses, that gives the resolution of 97,348. That means 1 lakh mass resolution is required by the instrument to separate 321.1513 and 321.1546. So that's where we can also, if you have the know the mass of the compound, so you can able to understand what the instrument required to separate the ions and based upon the resolution. So that if you calculate the resolution and 97,348 as of today, Orbitrap can able to separate these two because it is having a mass resolution of 1 lakh. So that's how we can able to use the mass spectrometers to separate closely associated masses. And exact mass determination is always needed to get the mass accuracy 5 ppm. This is the C19H15N14, which is having a formula weight of 201.1001 monoisotopic. That means there are no other isotopics. The mass accuracy will be calculated by using a formula called mass error divided by formula weight. This is the formula weight into 10 power 6. So mass error is equal to the mass accuracy, sorry, is equal to 5 ppm mass error. Sorry, this is mass error we are calculating. Mass accuracy is 5 ppm. That is the 5 ppm mass accuracy. Formula weight is 201.1001 by 10 power 6. So this formula we just change it to calculate the mass error. So this is the maximum mass error is allowed by a high resolution mass spectrometer to identify the mass of the compound. So in the mass spectrometers, there is a different type of scans are available. So one is a full scan. The full scan means we have a mass range. Suppose we are analyzing a small molecular weight. So what is the maximum molecular weight it can able to? So we have been starting with 50 low and to putting a 650 mass. So we can scan at this range to see that the compound will be identified. For many times this for unknown compounds will be used. And there is another process called selected ion monitoring, that is a SIM, and which is used mainly for the quantification. If you have a chromatographic technique hyphenated to single mass, we generally use single mass means we are not doing any fragmentation. We are only getting the single molecular ion. But if it is a GCMS will have an electron ionization, which can give the whole fragmentation in a single mass itself. So at which stage we will take the, generally we will take the base peak of the base peak in the mass spectra will be used to quantify the compound. So that is the reason SIM can be used. Only few characteristic ions are selected, increasing dwell time and sensitivity. The sensitivity will increase from full scan to selected ion monitoring to the other one is selected reaction monitoring. So this is nowadays people have been preferring because of the matrix interferences when we are handling bioanalysis exclusively, biological samples over SIM. SIM means that is we are monitoring one ion. Suppose we assume that we have a 44 and we have 4 different structures we have seen in molecular formulas. So when we select the 44 as the ion to quantify the compound, but 44 can be associated with other compounds also. So that time the quantification error can be possibly high. So this is another mode of technique, selected reaction monitoring we use, and for which we have to identify what could be the masses and fragments. So this is the electron ionization full scan spectrum. So this is the compound which is having a molecular weight of 182. And since EI can give a fragmentation in a single mass, so these are all fragments from it. So if you see 167, if the molecule is fragmented here, that means 182 minus 167. So 167, if you see here somewhere here the molecule, but it is not stabilized, it is immediately converted to 150Y. That means the fragmentation has occurred here. So like that, the mass will be looks like that if you are using EI. So if you are doing the compound in ESI ionization mode, if you see this is the compound 398.4. If you are using a single mass, we will get only molecular ion associated with some isotopic abundances. That's it. No fragmentation occur if you are using a single mass with ESI ionization. So the selected ion monitoring, there is a selected monitoring is just to detect one single ion. We are selecting one single ion, giving in the software that you have to monitor this ion. So that will be used for the quantification purpose. When we are using a single quarter pole, SIM will be the best to quantify the analyte based upon the single ion. Full scan monitoring is usually used to scan the whole range of ambiguous values, and it is usually qualitatively analysis with a limited sensitivity and selectivity. And SRM, selective reaction monitoring, MS detection with single ion filtering, then single product ion selected post fragmentation. This is usually when we are coupling MS with another MS. This is a triple quadrupole or QTOF or even exactive arbitrap, quadrupole and arbitrap. And there is another multiple reaction monitoring as SRM, but with multiple user-defined fragments. SRM and MRM almost both are same. So if you see here, this is the one chromogram TAC. Generally in a mass spectrometer, if you are coupling with any liquid chromatography or gas chromatography, that is chromatographic technique, we call it as a not a chromogram, it is a TIC, that is a total ion chromatograph. Because this is the peak, although it is there, but it is abundance of the ions collectively exist in the single peak. So that is called, this peak is called as a total ions of this EAM molecule. So like this total ion chromatograph. So if you see this is the same one sample having different compounds run in a full scan mode, and the same sample was run in a SIM mode. That means we have selected one single ion for each compound. So then you can see we can eliminate the noise successfully, and we can eliminate the interferences associated with the compound and the noise. So also we can improve the peak resolutions. So here, if you see here MDA, we can see a lot of peaks, other closely eluted peaks are associated, which can be eliminated by running the same sample in a selective ion monitoring mode. And the other one is selective reaction monitoring. If you're having a MS and coupled to another MS, this is one of the most important method to quantify the compound by using MS. So for which, when we want to do a selective reaction monitoring, suppose we are having one molecular ion, then if you do the MS, we will get the fragments. So what is the best fragmentation? That is, suppose if you have a 100 is the molecular ion, and 50 and 40 is the two fragments. So 100 to 50 is giving a very high intensity and sensitivity. Then we can select 100 converting it to 50 at a certain fixed collision energy. So that will select as a selective reaction monitoring. So for which initially when we run the single mass, we'll get the molecular ion. Then molecular ion, we select one molecular ion to further fragmentation, that is MS. So this ion will further fragmented at collision induced dissociation by using argon gas. And then these fragments we will see here, out of which this fragment converting to different fragments, which is the most intense peak, based upon which we select the product ion, which is arising from the precursor ion. This is called precursor ion, and these are all product ion. This is called product ion scanning. So when we want to do the product ion scanning in mass spectrometer, we have to understand what is the precursor ion. But even the reverse also can be possible. The precursor ion scanning can also be possible. If you want to understand, there are a lot of products have been forming here. So what is the precursor ion for this molecule? That can be possible if you give the product ion in the software and see that what is the precursor ion. So that also can be possible. So these are the one fragment precursor product ion is forming due to this precursor ion. So it will be given only precursor ion shown in the mass spectrometer for the ion selected as a product ion. And there is a neutral loss scanning, that means loss of water, loss of carbon dioxide, like that. So if one molecular ion converting to product ion, that means one precursor ion is converting to product ion, the loss, if it is 18 or 44, that means that is called a neutral loss scanning. So this also can able to see that what is the transition converting from precursor ion to product ion with a loss of neutral ion. So that also will give a information that what could be the best conversion of product precursor ion to product ion that can be selected as a selective reaction monitoring for quantification. So this is also possible multiple ion monitoring. Nowadays, people have been thinking about developing methods, multi-class analytes in a single method. So when we want to analyze many compounds in a single method, so we may have to use SRM or MRM as a quantification method. So here the precursor ion, we have selected after single MS, there is a fragmentation. Then we have taken the one ion and we have sent it to collision cell. So here the selected ion will further fragmented, and this fragmentation, which fragmentation gives the high intense peak here. Depending on that, we will select the transition from precursor to product as a SRM for quantification of that compound. So if you see, this is the spectra, which is having a product ion 399 is the precursor ion. So I want to find what is the product ions for 399. 399 is the molecular weight of this compound. This is the precursor ion. So I gave in the software what are the products for this. That means the fragments formed for this 399. If you see here, there are 381, 316, like that, 223. So out of this give a very high intense signal. That means the conversion of 399 to 223, that the fragmentation of 399 to 223 is giving a most sensitive for that compound. So we select 399 conversion to 223 as a SRM for the quantification of this compound. So that's how we select the SRM. So if you see, this is the one. Now in SRM mode, 399 to 223. So we will observe only single peak in the TAC. So why SRM is so special and why important? Because we can eliminate the noise in the protogram. And also we can selectively detect the respective compound of our interest. So it can give very high sensitivity with low detection limits and reduced sample preparation. Many times, even after using a best sample preparation techniques, but still some noise exists. That noise can be eliminated if you select the SRM or MRM mode for quantification. And consolidated analytic method and faster automated data processing can be possible. And it is a highly selective technique for quantification in LC-MS-MS or GC-MS-MS. And nowadays, now the people have been bringing this manufacturers are bringing a auto SRM overview where we can give a molecular ion. It can automatically select what is the energy required to convert precursor ion to product ion. So that is possible now auto SRM, and it can say that yes, this ion to this ion for this compound at this energy is good for the quantification. So that's how we can easily optimize the SRM process using the latest equipments. What is the advantage if you are performing a quantification by SIM and SRM, selective reaction monitoring? If you see, this is the para DDE at this concentration, same sample run in SIM mode as well as in SRM mode. If you see here, the compound being associated with other peaks also in the TAC. And also the sensitivity of the peak is reduced and the peak area, due to which the peak area is gone. So that means it is giving that means you have to use high concentrated sample to analyze the compound in SIM mode. But the same one, if you run it, there is no noise at all. Only single peak can observe, and with the peak symmetry and peak intensity and peak resolution is very high. So that's how this can eliminate the matrix interferences associated in the sample. So why is mass spectrometry such a good tool for these applications? Because of its selectivity with respect to the what type of the mass analyzer what we have been using, and sensitivity can routinely detect, analyze in nanogram per ml, picogram per ml, and even go up to femtogram per ml. And compatible with separation techniques like GC, LC, and UPLC, and even capillary electrophoresis, and robust and high throughput, and it is too versatile. And why mass spectrometry good tool for these applications? Because we can handle many type of matrices like biological samples and even food samples, and we can hyphenate even the sample preparation with LC-MS. Nowadays, online SPE with LC-MS. There have been hyphenations have been automated completely. That means you can, if you receive a sample, suppose if you receive a plasma sample, we can put it and we can program it the SPE process. Then go, after SPE, the eluent will directly go to the LC-MS. That is an automation method, and you can just go to your home and to next day if you come, your results are ready. So that sort of automation, that level of automation will giving a scope to use MS-MS more robustly in pharmaceutical analysis. And we have since full scan, SIM, many type of scans can be easily been used in different MSs. So tandem MS has increased the scope of use of mass spectrometers in drug development, drug discovery, even the drug safety evaluation and pharmacokinetics, and even the pharmacological aspects of the drug molecule. So at which here the sample detection is the one source which can use HPLC, GC, capillary electrophoresis as a sample detection sources. Ionization can be positively charged or negatively charged based upon which. So if you see that depending on the type of the analytes, we can choose what type of the sample detections can be used. Suppose we are analyzing the volatile molecules, then you can use gas chromatography, and we are now using non-volatiles, then we can use HPLC with ionization like ESI and MALDI. And even if you are performing the trace elemental analysis, that you can use inductively coupled plasma coupled with mass spectrometer. That can also be possible to analyze the trace elements in APIs, active pharmaceutical ingredients, and other matrices. But mass spectrometry is a powerful method to analyze the structure of organic compounds, but suffer three major limitations. That is, one is compounds cannot be characterized without clean samples. So matrix interference, biological sample and food sample analysis by mass spectrometer still needs some improvement. So this technique has not the ability to provide sensitive and selective analysis of complex mixture, For big molecules like peptides, spectra are very complex and very difficult to interpret. Sometimes even the like dioxins and furans also, we may have to separate before entering into the mass detector. So if you see here, the hyphenation has been increased. Hyphenation of the mass spectrometer with liquid chromatography, gas chromatography, capillary electrophoresis in inductively coupled plasma has enhanced the scope of the MS in drug development, pharmaceutical analysis. So we have been a lot of combinations are available like LC, QTOF, MS, quadrupole time of flight, LC Arbitrap, MS, gas chromatography with MS, and even gas chromatography with MSMS, and GC with quadrupole time of flight mass spec, like GC Arbitraf MS been recently introduced by Thermo Scientific, and LC with ICP-MS, and IC with ICP-MS, like that. So there are hyphenation has gained prominence because of to solve the complexity of the matrix interference, shorter analysis time and higher degree of automation, higher sample throughput and better reproducibility, improved selectivity and sensitivity, and can provide structural information of each separated compound. And hyphenation between chromatography and mass spectrometry has shown greater sensitivity than other hyphenated analytical techniques. So that is how this is a lot of advantages of hyphenating chromatography, that is a separation technique, with the identification system, that is mass spectrometry. So if you see GCMS proven to be the best hyphenated technique in biomedical and environmental analysis because it has advantages like identification. Even we can do identification based on the comparing the retention time of the analytes with the standard and then the sample, and also using the mass spectrum obtained by the mass spectrometer. And even fragmentation pattern of the compound and cheaper cost of analysis in comparison to the LCMS-MS, availability of spectral libraries, more resolving power than other instruments, and tends to have a high sensitivity. So this is the GC and this is the mass spectrometer hyphenated, and there is an interface. This is called transfer line because mass is operated in a vacuum and mass also have a gas phase chemistry. Basically mass is a gas phase chemistry it is operating. So we can easily hyphenate the gas chromatography with mass spectrometer without having much problem. Sorry. So here we are using a carrier gas. The carrier gas is here helium, that is the inert gas. The helium is the inert gas. So if you are using helium, then the sample will be injected in a liquid form at this injector. The helium gas will flow throughout the column. This column is called capillary column, which consists of a one stationary phase, and the mobile phase will be helium. So here the gas chromatography, we always use carrier gas, inert gas, because it should not react with the analytes what we have been analyzing. So here only helium for GCMS, that is the only option we have. We cannot use nitrogen, we cannot use argon in GCMS because here gas chromatography operates in atmospheric pressure and mass spectrometer operates in vacuum. So we are using a high density gases, then we need to have a big vacuum pumps to maintain the vacuum in mass spectrometer. So that is the reason helium is the less dense than any other inert gas. So there is no other option till now. But now people have been facing issue with the helium cylinders because availability of helium is becoming a too scarce now. So that's the reason now the manufacturers have been coming with alternate gases like helium with mixing with nitrogen or something like argon, like that. And sometimes people are using only nitrogen with some arrangements and modifications in the interface what we have been using. So here there is a oven. So this is the temperature programming used. The ions, when it enters into an injector in a liquid form, we are putting some high temperature here, and this all molecules enter into the gas chromatography in liquid form will convert into gaseous phase. So then these gaseous phase ions will be gaseous phase compounds will be separated by partitioning of the analytes between stationary phase and mobile phase. And then the separated compounds will go to the mass spectrometer. The mass spectrometer contains one is the ionization source, mass analyzer, then the detector. So the ionization source, once the compound separated, the compound enters into here, the ionization fragmentation, because we are using a electron ionization as a ionization source in GCMS. So here the ionization fragmentation simultaneously occur. So these fragmented ions will go to mass analyzer where the separation of ions will happen, then goes to the detector where we can detect the compounds, detect the ions. So that's how GCMS works. So here the transfer line temperature, we maintain always temperature equivalent to the source temperature, what we have been putting. That is a less than 50 of the source will be put as, more than 50 of the ion source will put transfer line. Because ion, this here the molecules are coming in a gaseous phase, they should not condense here. If the temperature is low, there is a chance of condensing these ions here itself. So in order to avoid that, we are using a temperature that is called transfer line temperature. We always put in a GCMS. So this is how the GCMS is a old GCMS. Now it has been too modified and it has becoming a very small. In this GC also, people have been designed GCMS nowadays. And this is the carrier gas where we have been coming with the helium we are using, and some traps like we have to remove any impurities like hydrocarbons, moisture, oxygen in the helium gas. So for which we are using a traps here. So GC always have a quarter pole, also can be possibility of GC mass. Mass contains a mass analyzer of quarter pole, triple quarter pole, even QTOF, and people now Therma came with a GC with Arbitraf mass analyzer. So this quarter pole is basically looks like here 4 magnetic racks, and we have been connected with the DC current. That is, so we are creating an electromagnetic radiation. So once the ions after the fragmentation at the source will enter into the mass analyzer. So based on the mass by charge, these ions will be separated here. And when we are giving in the software, suppose we want to monitor only ions in 200 to 400, that is the mass range. So at this mass range, so if you want to separate, then this electromagnetic radiation is created only in that mass range. So at in that mass range, so the ions below 200 and above 400 will be thrown out from this quarter pole mass analyzer through to a venting program. Only the ions between 200 to 400 will enter into the mass analyzer to reach to the detector. So that's how the masses will be separated by using this. So this is the real picture of the quarter pole, how it looks like. This is 4 magnetic rods, and we have a copper wire DC current here flowing. So it will create a magnetic electromagnetic radiation, which is specific to the ion current. So if you see the GCMS analysis, this is the analysis of the dimethyl organophosphorus metabolites in urine sample. So we can see the good separation, and also simultaneously we'll get the spectra. So and we have GCMS has libraries. We can compare the spectra, this fragmentation pattern with the library. So it can easily able to identify the compounds. That's the reason simple GC with a single mass itself, it can solve the many type of analysis. But when it comes to the liquid chromatography mass spectrometer, LC-MS has proven to be one of the best technique in bioanalysis, pharmacokinetics, and all in basically in drug discovery and drug aspect. It has advantages. It also can able to compare the retention times of the analyte with the standard of that purified version of that compound, and also based on the mass. Fragmentation pattern can be possible if you have a LC with tandem MS, LC, MS. And availability of spectral libraries. Nowadays, manufacturers have been designing and designing the libraries we can able to use for many of the compounds, which established compounds. More resolving power than other instruments, because we can able to couple these high resolution mass spectrometers with LC very easily, and tends to have a sensitivity and ability to analyze biomolecules as well as small molecules. The disadvantage of one gas chromatography is that we can analyze only volatile compounds. We cannot analyze compounds with having a polar functional groups directly without some additional steps. But LC-MS can able to do that without any derivatization process and all. We can analyze polar molecules by using LC-MS. So the components here is only inlet system. The inlet system is here liquid chromatography. Then rest of this mass spectrometer components. This is how the schematic representation of the LC-MS. So this is the pump which has been pumping the mobile phase throughout the system. Then there is a sample, multi-component sampler where we inject the sample here in liquid form. So this is the column. This is the column have packed with the stationary phase that will separate the molecules, and then the separated molecules through an interface. This is the interface, because here it is the sample is coming in a liquid form, and it has to enter into mass which is operating in a vacuum. So for which we cannot directly place the liquid into the mass spectrometer. So this interface has been designed in such a way that the molecule will enter into a nebulization through a process called nebulization as a aerosol droplets. That is called spray, electrospray or atmospheric pressure spray. So this is how the interface will be designed to use the mass spectrometer easily with hyphenating with liquid chromatography. So this has a mobile phase, it is flowing throughout the system. And here the column stationary phase, when we inject the sample here in a liquid form, it directly, the mobile phase will take the sample directly into the column. Here the separation of the compounds from the sample will occur. The separated compounds will go to mass to detect the molecular ion, then fragmentation, then for quantification. So LC ionization techniques have also revolutionized quantitative MS exclusively in bioanalysis like pharmacokinetics and pharmacodynamic testing. Ionization is required for mass spectrometry, and GCMS applications are limited to volatile and thermally stable compounds. APCA, ESI, and APCA removed these limitations, allowing LCMS applications to expand to a wider variety of compounds. So if you see the wide range means even we can analyze small molecules, and also we can analyze larger molecules like proteins, peptides, oligonucleotides, RNAs, lipids, biomarkers, proteins by using LCMS. And LCMS, when it comes to the pharmaceutical research, impurity profiling has gained prominence, and LCMS can play a wide role in characterizing the newer impurities as well as quantifying the impurities associated with the active pharmaceutical ingredient. So this is how, when we want to characterize the impurities, so this is the steps we have to follow to derive a final structure. So you can refer the ICH Q6A document for more details about the impurity profiling. So if you see here NDMA, nowadays people have been talking about nitrosamine impurities in ranitidine. There is a lot of issue and havoc for the presence of this NDMA, which is highly carcinogenic compound in ranitidine tablets, which have been routinely using by many of us. So that is the level of this is very low, but it is since FDA says that it should not at all detect. So they have been fixed some LOD value 0.3 nanogram. So we also developed a method using this mobile phase and all, and we could able to detect even using a triple quadrupole MS. They have been initially published the LC Arbitrap MS method, but later on that they have also came with LC triple quadrupole. So we developed a method by the time, and we analyzed many samples from industry for the presence of NDMA in ranitidine. And even LC-MS also has a plays a wide role in bioanalysis. Bioanalysis is the subdiscipline of analytical chemistry, which covers quantitative measurement of xenobiotics and biotics. So we can quantify one is the separation of the analytes by using a liquid chromatography technique, then identification, then detection, then quantification. So there's all 4 pages can be done with a simple LC-MS. So simple is methyl malonic acid. This is a one marker compound. If this MMA level is high in plasma, that means the person which has found MMA levels high means he is deficient of vitamin B12. So to monitor such type of clinical diagnostic analysis, where LCMS plays a crucial role for this type of compounds. So this is one application of LCMS. And also, if you want to determine the drug molecules, suppose if you run a lovastatin, simvastatin, they are very closely associated. So if you run in sim mode, they're almost very close to each other. So separation may not be possible. But same if you run in SRM or MRM mode, you can get a very good sensitivity and good accuracy in the quantification of those compounds in API samples. So if you see here the application range of GCMS versus LCMS, GCMS is very limited because it is limited to volatiles and semi-volatiles. Some non-volatiles like polar compounds can be analyzed, provided if you can derivatize them by using a process called derivatization with silylation or acetylation or alkyl chloroformate derivatization. But if LCMS with ESI and APCA sources, we can cover many range, almost non-ionic to ionic. So that's the reason LCMS has a very broad application in pharmaceutical analysis. So I'm coming to the final stage of the talk, summary. The summary is mass spectrometry is a powerful analytical technique in pharmaceutical research due to its selectivity, sensitivity, resolution, and also precision and accuracy in quantification, and also ability to hyphenate with other chromatography techniques very easily. So that makes it is the best technique to use in pharmaceutical research. So this, I am very much thankful to you all, and thanks to Vishwanathan sir for giving opportunity to deliver my talk at your podium. Thank you so much.
Thank you, sir.
Yeah, any questions? I can take it, no problem.
Sir, we have few questions.
Yeah, please.
I'll ask you.
Go ahead, go ahead.
Okay, sir. Swapna Verma, she asks, which mass analyzer is advanced and why?
Which mass analyzer?
Which mass analyzer is advanced and why?
Oh, okay. Mass analyzers, I think nowadays ion cyclotron resonance is advanced version. But as such, if you consider the pharmaceutical research, Arbitrap is highly advanced now because of its resolution at 1 lakh. That means it even very closest of the closest masses of the compounds can easily separate it by using Arbitrap mass spectrometer.
Okay, sir. We
Yeah.
have one more question.
Hmm.
Can I ask?
Yeah, please go ahead.
Can we differentiate the peak between one isotope and metabolite of other compound?
Yes, yes, it can be easily discriminated if you are using high resolution mass spectrometer.
Okay, sir. And one more question from Dr. Amartya De.
Hmm.
Why same compound show peak at different retention times during GCMS analysis?
Same compounds show peak at different retention
Peak
time.
at different retention times.
So that can be possibly due to the isomers. Suppose if you have a hexachlorohexane, that is a pesticide, which exists in 4 different isomers: alpha, beta, gamma, and delta. So
Okay.
that can be separated because we are GC is a basically separation technique. If you are using a high resolution stationary phase, the stationary phases which can able to even separate the even chiral molecules also can easily be separated.
Okay.
So if you see the chiral molecules are isomers which having a same mass, same masses, but you have a due to the partitioning of the analyte between the stationary phase and mobile phase can be separated. So that's the reason hyphenation of GC and LC with mass will able to give this type of chances. So alpha, beta, gamma, delta, HCS will come in a different retention times, but will have a same mass. That is 191, I think 217, something like that, same mass. So that can be possible.
Okay, sir. And we have one more question, sir, from Dev Prasad Ghosh. Can any two different molecules have same fractions? Then how to discriminate?
Can you read again?
Can any two different molecules have same fractions? Then how to discriminate?
Can two different molecules
Have same fractions.
Same fraction means suppose if you are collecting the sample in different fractions, that's what?
I think so, sir.
I'm a little confused, but suppose if you have a same mass in a different fractions also can be easily discriminate, and we can even quantify that how much compound present in that each fraction by using mass spectrometer hyphenated to LC. That is possible.
Okay, sir. Thank you. Questions are over, sir.
Okay.
My sincere thanks to Dr. Mohan Krishna Reddy Garu for accepting our invitation and presenting a nice information on mass spectrometry and its importance in pharmaceutical research, in drug development and all. And we are very thank you so much, sir, from our management and the staff and from the chairman of Vignan Pharmacy College. Very thank you, sir.
Thank you. Thank you so much.
Thank you, sir.
So shall I leave now? Okay.
Okay, sir, you can leave now.
Right. Okay.
And thanks to all the participants who have attended the session. Thank you.